Magandang umaga po sa ating lahat. Welcome to the Filipino Science Hub. Thank you for your continued support to this online platform. To our new participants, thank you for checking us out today, and we would like to officially welcome you to the Philsci Hub family. So bago po tayo magsimula, I would like to share some house rules in order to facilitate a smooth session for everyone. For our Zoom participants, we would appreciate it if you could turn your audio and video cameras off during the webinar. If you have questions or comments, feel free to type them on our Zoom chat box or YouTube live comments, and we will try to accommodate as much questions and comments as possible. After the talk, we will have a 30-minute Q&A where the Zoom participants will have the opportunity to ask their questions live. We would also like to request for everyone to not record the webinar. The full video of the session will be posted on our official YouTube channel. For those who would like to receive a certificate of participation, a Google form link will be posted on our Zoom chat box, YouTube live comments, and YouTube video description towards the end of the webinar. Please make sure that you enter the correct information, especially your email address, to ensure receipt of the certificate. Also, for you to obtain the certificate, you will need to subscribe to our YouTube channel as well as follow our page on Facebook. Please note that we receive hundreds of requests and it takes around 7 days or so to process all the certificates. If you do not receive your certificate after a week, please feel free to send us a message on Facebook or email us at philscihub@gmail.com. So again, welcome po sa Philsci Hub. Welcome to all our Zoom participants and our YouTube live viewers. For those who are new and may not be familiar with who we are and what we do, allow me to share our mission and vision in the next few minutes. Filipino Science Hub is an online platform founded by our CEO, Filipino scientist Dr. Jeffrey Camacho Bungkin, back in 2012. Currently, we are a registered nonprofit organization based in Houston, Texas, United States. Our mission is to promote STEM education and the culture of research among students and teachers in the Philippines and abroad. Our current global leadership team is comprised of volunteer Filipino scientists and seasoned STEM educators from different parts of the world. Our vision is to see a technology and innovation-driven Philippines, and we believe that this can be achieved by creating a new generation of well-rounded Filipino STEM enthusiasts. This vision is being carried out via our two major programs, the Philsci Hub Ed program and Philsci Hub Research University. Through Philsci Hub Ed, we are able to empower and support our teachers when it comes to strengthening STEM fundamentals education. We believe that this is important because the more equipped our STEM educators are, the more they can inspire our students to pursue a career in science, technology, engineering, and mathematics. Through this program, we have been able to serve our teachers by creating STEM teaching modules as well as conducting teachers training webinars, all of which are accessible on our website for free. So we have also been able to collaborate with major educational organizations that have the same goals and advocacies as Filipino Science Hub. Through these partnerships, we have been able to further expand our reach and help more students and teachers, especially during the time of pandemic. So together, everybody achieves more. The other major program that drives our vision forward is Philsci Hub Research University. We believe that in order to strengthen a STEM culture, students also need to be able to learn how to conduct a scientific research. As we know, many scientific and technological breakthroughs happened through research, and we would like to equip our younger generation by providing them proper scientific research training. So ika nga po, start them young. Here at Philsci Hub Research University, we provide free courses on fundamentals of scientific research, such as research ideation, literature review, how to write a research proposal, how to communicate your research work to the scientific community, among other courses. Through this program, we are also able to bring practicing scientists closer to the STEM educational sector. So because of this closer contact, our students become more aware of the possible STEM career paths available for them and how these careers contribute to the advancement of the society around them. So up to date, we have been able to reach over 70,000 students and teachers all over the world. And we know that this will continue to grow in the years to come. And this is why we are all very excited on what is in store here at FSH. Our official website is www.filsihub.com. This is where you can access all our materials such as webinars, tutorials, modules, virtual lab, and special features. We are also present in several social media platforms such as Facebook, LinkedIn, YouTube, and TikTok. So follow us wherever you're most active on to get updated with our web events. So again, thank you for being with us this morning. And to officially start our webinar, I would like to turn the virtual floor over to our host for today.
Ayan, magandang umaga po sa inyong lahat sa Pilipinas. I would like to call on my co-facilitators for today, so Janice and Mama Connie. Ayan, so magandang umaga po sa lahat ng nasa Pilipinas. I think Mama Connie is still muted. Alright, so ayan, magandang umaga po sa inyo lahat. Magsisimula po kami sa pamamagitan ng pagpapakilala. So ako po si Jeffrey Buntin and I am the founder of Filipino Science Hub. I am a Filipino chemist and scientist based here in Houston, Texas. So masaya po kami na makasama kayo. So we have about 300 live participants today on Zoom and on YouTube. Alright, Janice?
Hello, ako po si Janice. Ako naman po ay based in Kansas City, Kansas, USA. Ako naman po ay ang secretary ng Filipino Science Hub and in charge sa communications ng organization namin. And we're very happy po. Welcome sa aming webinar for today, yung first installment ng ating Biology Month.
Yeah, and speaking of... Oo. And speaking of, teka, bago natin ipasok si Mama Connie, siya kasi yung pasimuno dito eh. Siya kasi yung pasimuno dito. So like, by the way, everyone, to all the students and educators out there who are tuned in, itong Biology Month was actually the brainchild of Mama Connie, who happens to be the very first biologist and a world-class Filipino scientist in the areas of taxonomy and mycology. Siya yung kauna-unahan nating biologist sa PhilSci Hub. And she came up with this. So Mama Connie, why did we come up with this again?
Because we want everyone to be a biologist.
Yan. So,
recruitment pala.
Yeah, one of the main objectives of Biology Month is to give students an idea of what it is like to pursue a career in biology, di ba, Mama? So, specifically, the way we designed this program is such that mapapakita namin sa inyo ang fundamentals of biology and then all the way up to its applications, yung mga practical applications ng biology. And so that's the reason why we came up with this webinar. And would you actually believe that we've been doing web events for about 2 years na, no, Jaja?
Ever since nag-start yung pandemic.
So 2 years na po kami puyat every Friday dito sa US. We also have members dialing in from Europe actually. And this is, I think, our 63rd web event. So would you believe? So parang more than with about 52 weeks in a year. So more than 1 year natin tong ginagawa. And this time, okay, so di ba napachika na ako, hindi ko na na-share yung aking slide. Ayan. So, but I will be sharing. Let me share my screen first. Ayan. So, Biology Month, we will have a panel of leading Filipino scientists in different areas of biology. So, this is going to be our first installment, but this session is extra special because, Jaja,
Wait lang. Sorry, ayan. Special, nawala pala. Special shout out sa ating STEM students na participants for today from Bataan National High School. These are senior high school students from Balanga, Bataan. And thank you din pala sa teacher nila kay Sir Jay sa pagpapa-attend nitong mga estudyante natin ngayon.
Yeah, makwento ko lang. So we would just like to commend Sir Jay kasi he reached out to me kasi gusto niya talaga ma-experience to ng students niya. So, sir, maraming salamat po. Jaja?
And you are in the right place.
Yes.
You are in the right place. Ayan. So, para po ipakilala na yung ating special speaker for today, let us also... Ay, sorry. Sorry. Baliktad. Ayan, sorry.
No problem.
Meron muna pala tayong pre-test bago ang pagkakilala sa ating speaker. So, ayan.
So sa mga students who have dialed in, we'll be sharing a link to a very short pre-test po. And this is for a way for us to be able to gauge kung paano ba yung impact ng content po. So these are just 4 questions. We're gonna give you a couple of minutes to answer these questions, and we'll be sharing the pre-test link sa Zoom chat box and also sa YouTube channel natin.
Ayan, we will be giving you like maybe 2 or 3 minutes para masagutan yung pre-test natin. Kasi favorite natin tong classroom. Paano tayo nasa classroom ngayon lecture hall, kumbaga.
Yes.
Meron muna tayong pre-test.
Yeah, so we have more than 300 live participants today. So we're glad to have everyone in the session, and we hope that you can actually you not only learn the technical lessons when it comes to the today's topic, but hopefully yung sense of inspiration rin hopefully ma-convince namin kayo to consider biology as a potential career area that you would pursue after senior high school.
Ayan, posted na po both in Zoom chat tsaka sa YouTube natin, YouTube chat ang link para sa Google Form, para sa pre-test natin. Ayan, kayang-kaya niyo yan. Pero don't feel bad kung hindi niyo pa alam ang sagot. Kasi
pre-test to. So it's okay if you don't know the answer. Yeah, but our goal, hopefully, after the session, hopefully you listen intently and then masagot nyo yung mga tanong after niyan. Ayan. So, Janice, ano ba ang mga recollections mo ng iyong high school biology?
High school biology? Teka.
On the spot lang.
Oo, on the spot ako. Parang hindi nakikinig sa teacher.
Ayan.
Parang may mga, ano ba yung naalala ko? Parang wala.
Hindi mo hinayaang...
Chem kasi talaga yung favorite ko nun eh.
Ayan.
Hindi mo hinayaang lumakad yung palaka na hindi pa nasara yan dyan?
Parang wala kaming palaka.
Hindi kayo nagda-dissect?
Ang recollection ko ng biology class ko ay sa college na.
Doon po kasi mama sabi.
Doon kasi kami nag-dissect talaga ng palaka. Na-enjoy ko naman siya.
Yan. Alright. Ako naman, wala kasi kaming lab ng biology nung high school kasi 19 kopong-kopong pa. Hindi. So medyo nasa 90s pa yun, 1999 yata. Di ba? So, alright. So, let's see. Alright. So meron din tayong participants. Yeah, we have a participant from Tsukuba City, Japan. So,
wow.
Si Myra Villarreal. Yan. And then, also from Isabela State University, si Mr. Mike Tabug. So, ayan. So, kung tapos nyo na po, sagutan yung pre-test, could you mag-thumbs up nga kayo sa Zoom para alam namin kung tapos na kayo? Are we good?
Ayan, may mga nagta-thumbs up na.
May mga mga yan.
May mga bibo tayo ngayon.
Oo, ginagalingan naman. That's good. That's good. All right. So I think that should be enough time, para sa pre-test. So all right. So we're moving on. Pupunta naman tayo sa introduction na.
Ito na ba to talaga?
Yeah, ito na to.
Ito na ba to?
Yeah, and to introduce our speaker for today, we will have Dr. Connie Gibas.
Okay. I'm on, right?
Yep.
Okay, hello, Hubbers, as we say in Texas.
Yep.
Hi, y'all. Alam niyo yun. Ganon yon dito sa Texas. Anyway, April is Biology Month. Sinabi na to ni Dr. Ja. So our succeeding webinars will be on anything biology. So our first speaker for this month is Dr. Marilen Balolong. She is a professor at the Department of Biology, College of Arts and Sciences at the University of the Philippines, Manila. So Dr. Balolong and I go way back at the University of the Philippines at Los Baños at Biotech. She worked as one of the research associates at the Microbial Culture Collection and Services. And before she became Dr. Len Balolong, we knew her as Len Len. As you know, the name is very popular these days. But anyway, Dr. Balolong is very accomplished in her field. She did her bachelor's and master's degrees in microbiology at the University of the Philippines, Los Baños, and her doctorate in public health at UP Manila. She did a postdoctoral fellowship at Dankook University in Korea, and she has 50 publications and 3 chapters in a book to date. She has received numerous awards that include several international publication awards, 3 Gilles Pico Award as one of the University of the Philippines top scientists. In 2018, she was recognized as one of ASEAN's outstanding science diplomats by the ASEAN Foundation and Harvard Kennedy School. So now I present to you Dr. Balolong.
Morning, everyone. Am I on?
Yes po.
Yes, hi. Good morning. Good morning. Thank you, Tita Cons, Mama Cons, for that lovely introduction. And I'm really getting giddy because it's more than 300 right now. No, I'm so excited, and I'm sure there's quite a lot of youth and the other guests from the live stream. Magandang umaga po sa inyong lahat. Ako ay excited na magkwento sa inyo, hindi lang tungkol sa sagot sa quiz kanina, kundi tungkol din sa mga many things that na mga ginagawa ko. So today, I'm really very honored for the invitation, Phil-Sci Hub. This is really a passion and advocacy of mine to educate and prepare students while they're young, to be involved with STEM. And this is my own little way of doing that by saying yes to the invitation of Phil-Sci Hub. So maraming salamat for this platform and maraming salamat for the opportunity. So for this morning, ang i-share ko po sa inyong lahat ay tungkol sa bacteria. Hindi ko alam kung marami na sa inyo ang nakarinig about the bacteria o kaya first time nyo itong narinig. This is all about evolving strategies to identify and functionally profile bacteria from communities. Ang daming technical terms, pero ipapaliwanag natin isa-isa. Ano ba yung identify? Ano ba yung functionally profile? At ano ba yung mga community? And particularly, I'll be dealing with applications for health and environment kasi yan po ang tema o doon umiikot ang trabaho ko sa University of the Philippines, Manila. So before ko ikwento, sige next slide, Jeff. Before ko ikwento yung mga topics, magkukwento muna ako ng aking microbiology journey. Kasi baka yung iba sa inyo nag-iisip right now na ano kayang gusto ko paglaki ko, kapareho pa ba ito nung nasabi nyo nung kinder, nung umakit kayo sa stage ng what I want to be when I grow up, o gusto nyong mabago. So bibigyan ko kayo ng option being a microbiologist. So kukwento ko yung aking journey. And then after that, I'll be talking about some topics related to my talk, which is microbial world. Ang emphasis ko dito ay bacteria. Madami pa pong mikrobyo, pero aabutin tayo ng mga 1 linggo kung pagkukwentuhan natin sila lahat. So bacteria po muna tayo ngayon. Ano? And then bakit ba natin pinag-aaralan itong mga ito? Various ways on studying them. Meron tayong mga traditional or routine, no? O yung mga unang panahon, ito na yung ginagawa nila, no? At meron din tayong mga advances, no? Papakita ko rin sa inyo yon. And then, syempre, sa bandang huli, no? Iyayabang ko naman yung mga miyembro ng aking laboratory mula sa aking mga estudyante at mga RAs na nag-go microbiology, no? At ito yung kanilang mga ginagawa. So, the last will be some sort of an invitation so that you could join us, no, in this advocacy. No? Sige, next slide po, Sir Jeff. Ayan, sige. So, as a microbiologist po, ang goal ko talaga personally ay to help improve the lives of the Filipinos, no? Kaya po yung aking costume ngayon ay lagit-lagi para sa bayan, no? Kung ako ay isang teacher, ang iniisip ko palagi, paano ko mapapagsilbihan yung aking mga estudyante, kung ako ay isang researcher, paano ko magagamit yung aking mga proyekto sa ikauunlad ng ating mga Pilipino, at syempre, pagdating sa public service, ako ay isang Rotarian, at ginagamit ko ang posisyon na ito para makatulong, no, sa ating bansa at sa ating kapwa Pilipino. So hindi ko ikukwento ang aking mga iba't ibang costume na ito, no, at iba't ibang liga. Ikukwento ko ngayon ang aking research. Ako po ay isang siyentista or isang scientist na nagtatrabaho sa UP Manila, at medyo malawak na rin, no, yung karanasan dahil iba-iba na yung aking mga na-try, no, o nasubukan na research topics. So ito yung ikukwento ko sa inyo ngayon. No? Next slide, Jeff. Yan. So, syempre, bilang isang scientist, kailangan mo munang malinang or ma-mold, maturuan, no, o ma-mentor ng mga experts sa iyong larangan. At nagpapasalamat ako at nabigyan ako ng opportunity ng University of the Philippines, Los Baños, particularly ang Microbiology Division. Diyan ako nagtapos ng aking BS at saka ng MS. So, ang laki ng pasasalamat ko at ako ay nahawakan, no, ng mga haligi ng microbiology ng ating bansa. That's why yung mga natutunan ko sa kanila, no, yan yung ina-apply ko, no, sa aking mga estudyante, no. Minsan nga pag gino-joke ko yung mga teacher ko dati, ang natutunan ko po sa inyo ay paano magpa-exam ng mahirap. Pero syempre joke lang po 'yon, ang pinakamahalaga ko siya talagang natutunan ay ang discipline, no? Isa yang pinaka-importante, at ang pinakamahalaga ay ano ba yung value? Ano ba yung value? Bakit mo ba ginagawa ito, At ang lahat ng 'yan ay nadala ko paglipat ko sa University of the Philippines, Manila, Sa UP Manila ako ay nagturo at nabigyan din ng opportunity para makapag-doctorate sa University of the Philippines, Manila College of Public Health. So isa sila sa pinakamagaling na public health institutions, hindi lang sa Pilipinas, kundi sa buong region. Dahil dito, hindi na lang ako nag-aaral tungkol sa mga mikrobyo, kundi nag-aaral na ako sa mikrobyo para sa ikagagaling ng publiko. So I'm a microbiologist for public health, Sa alang-alang na natin ngayon, yung populasyon, yung community, hindi na lang yung mga mikrobyo, Okay, next slide, please. So, ikukwento ko sa inyo, bago ko kwento yung yung mga basics ng bacteria, kasi dito nyo makikita dun sa nangyari sa buhay ko, kung bakit itong topic na ito ang gusto kong ikwento sa iyo ngayong araw nito, So my first love interest talaga ay mga fungi, di ba, Mama Cons? Kasi yan po talagang inassign sa akin, nung ako ay research associate pa lang. So whether I like it or not, I really have to appreciate the fungi. Kung familiar po kayo sa mga kabute, kung kayo ay kumakain ng mga mushrooms, yun po ay isang klase ng fungi, And I'm also very blessed, no, and lucky to be handled and mentored, no, by no other than Dr. Kevin Hyde, a very famous taxonomist, no, of fungi, and of course, Dr. Steve Pointing, isa naman po siyang sikat, no, at napakagaling din na fungal biotechnologist. So sa kanila po akong dalawa, no, nalinang tungkol paano ba aralin ang mga fungi, ano? And through time, nakita ko yung evolution ng pag-aaral ko sa fungi from taxonomy, fungal biotechnology, at ngayon, no, inaaral na namin sila using advanced technologies. Kasama ko naman ngayon yung aking mga estudyante. No? Next slide, please. So sabi nga nila, minsan yung first love mo, hindi yun yung nakakatuluyan mo, di ba? Alam ko maraming nakaka-relate dyan, no? So my heart did not stay with fungi. Instead, my heart stayed with lactic acid bacteria. So ito po yung tinatawag ko na my lab life. So marami po akong natutunan tungkol sa mikrobyo na to. Ito po ay nakikita sa mga fermented foods kung kayo ay mahilig sa yogurt, kimchi, burong mustasa, at kung ano-ano pa. Yan po ang bahay ng mga mikrobyo na gusto kong aralin through time. No? Marami na rin po akong nailathala na publications kasama ng aking mga estudyante, colleagues, no, and mentors tungkol sa lactic acid bacteria. Isa po dito ang slide na to, and next slide please. At pati po yung mga book chapters and reviews tungkol dito. Kinakabit ko na rin ang pag-aaral ng lactic acid bacteria sa gut microorganisms o yung mga mikrobyo natin sa tiyan na tumutulong sa atin paano i-digest yung ating mga kinakain. Kasi alam nyo ba na yung mga kinakain natin at ito yung nag-fuel sa mga mikrobyo sa tiyan natin at nagdidikta kung ikaw ay magiging malusog or hindi. So yan po yung mga inaaral ko ngayon. Ano? Next slide please. Nagsimula din ako at ang aking mga estudyante. Pasensya na ko, lagi kong hinihila yung aking mga estudyante kasi ang gagaling po nila, kaya lagi kong hinihila sila. No? So, nag-umpisa kami na inaaral lang namin mga set or grupo ng mga mikrobyo. No? Nagsimula kaming aralin yung mga mutations ng Mycobacterium, pinagkukos po ng TB. Alam nyo ba na yung TB natin ngayon ay marami ng antimicrobial resistant? Ibig sabihin, kahit gamot kayo ng gamot, hindi gumagaling, no? So, may mga set po ako ng mga estudyante na inaral yung mga mutations na yun. Bakit sila naging resistant, ano? Ibang topic ito at nako, happy ako na mag-talk ulit tungkol sa antimicrobial resistance in another schedule. Meron din kaming, naalala niyo ba yung undoy? Nung mga taga Maynila, grabe kung paano tayo pinahirapan ni Undoy, no? Nung unang moment din na yon, yung mga estudyante ko po ay nakaisip ng tingnan natin bakit nagkaroon ng leptospirosis case sa Marikina. Dati po walang lepto sa Marikina, and because of that event, no, dahil tumaas ang tubig, nagbaha yung buong municipality, tinrace po ng mga estudyante ko yung paano nakapunta sa bahay-bahay, yung mikrobyo. No? Nag-aral din kami tungkol sa actinomycetes. Ito naman ay mga grupo ng mikrobyo na nagpo-produce ng napakahalagang metabolite or compound para maging magamit na alternative sa antibiotics o kaya maging antibiotics. No? At ang pinakamahalaga, I think, at nakakakilig na bahagi, no, ng aming journey ng ilan kong estudyante, ang pag-aaral sa sa isang bacteria, no, ang Strenocomonas, ano. Ito ay dati kasing Pseudomonas ang pangalan niya, tapos nabago yung pangalan niya, no. Nakita namin ito sa mining environment at kaya niyang i-resist ang heavy metals. At nakakakilig po ito kasi yung kanilang photo, no, ng kanilang mikrobyo na nakita doon ay ginawang cover ng Philippine Journal of Science issue. Kaya tuwang-tuwa po ako. So, itong mga set ng una kong mga estudyante, this is around 2004 hanggang 2010, ang inaaral lang namin ay mga tig-iisang mga mikrobyo. Mycobacterium ba yan? Leptospirosis ba yan? Actinomyces ba yan? Pseudomonas ba yan? Ano? Pero nung nag-evolve ang mga bagay-bagay, nag-evolve din po, no, ang aming mga interest. Next slide, please. So from studying colonies, we now venture on microbial communities or consortium. At from studying just their itsura at paano sila i-describe, no? Inaral na rin po namin ang sequencing technologies. No? So halimbawa, sa halip na aaralin mo lang ang isang mikrobyo sa ngipin, inaral namin yung buong oral microbiota. Imagine niyo lahat ng populasyon sa loob ng inyong bunganga, ngipin, dila, saliva, gilagid, at kung ano-ano pa. Inaral po yan ng aming mga estudyante in partnership, no, with dentists. No? So nalaman namin at na-profile yung mga Filipino, kung meron ba kayong tooth decay or wala. Ano ang mikrobyo sa loob ng inyong bunganga, no? And then also, hindi lang sa health, meron din kami sa environment. Inaral naman namin ang microbial community sa aquaculture. So, kinumpara namin yung mga merong fish pen at merong walang fish pen at tinignan namin anong relasyon nila sa health ng mga tilapia, no? So, this is a really very exciting changes, no, after 2010 kasi ito na yung nakikipagsabayan na po tayo, no, ang Pilipinas, so particularly our university, no, ay nakikipagsabayan na sa ibang bansa tungkol sa mga ganitong technologies. Next slide please.
It's 11 o'clock.
Ayan, okay. And then, ayan, nabigyan po tayo ng opportunity para mag-aral ng makabagong technology sa Korea. So during my doctorate, nabigyan ako ng opportunity to do a sandwich program, gawin yung thesis ko doon, at magpatuloy ng postdoc. Doon ko po natutunan ang next generation sequencing. No? Nung nag-aaral kami noon, wala pa yan sa Pilipinas. No? Hindi pa nila alam yan. So I said, bakit hindi ko kaya iuwi ito? No? Iuwi natin ito, tingnan natin yung technology, kung kaya natin,
Dito
sure,
ka enrolled
ng Pilipino, no? So kayang-kaya natin ito, ano? At dahil dyan, meron kaming isang set at grupo ng estudyante, yung number 1 na publication dyan, ano? Ito po ay gawa sa UP Manila, no? At ito ay next generation sequencing. Ito ay 2017. Ibig sabihin, kayang-kaya, di ba? So sa ngayon, kami po ay nag-advocate ng paggamit ng next generation sequencing, katulong at katuwang ng Philippine Genome Center. Tinutulungan nila po kami para ma-afford namin ang next generation sequencing for health and for environment research. Next slide, please. Ayan. So yun po yung journey ko, no? So bakit ko nakwento yun, ano? Kasi kung mapapansin nyo, ganun din yung magiging daloy ng aking kwento tungkol sa topic, no? So mag-uumpisa tayo sa inaaral natin ng mga mikrobyo ng isa-isa lang. Inaaral natin sila tungkol sa itsura, no? And then later, aaralin natin sila collectively, no? Community-wise at using advanced techniques. No? Yan po yung topic natin ngayong araw nito. Okay? So, let's start with this slide. No? Sabi ko nga kanina, napakarami pong mikrobyo. Ano? So, imagine the 3 domains of life. Hindi ko alam kung na sumagi na ito sa lessons ng junior and senior high. Ano? Pero tayo po ay nakakategorize sa 3 domain. Ang mga human beings ay kasama dito sa eukaryota. No? At ang mga mikrobyo ay nakadistribute. Sila ay pwedeng maging archaea, sila ay pwedeng maging eubacteria, at ang ilan ay nababahagi or nakakategorize pa rin sa eukaryota. No? Pero dahil in the interest of time, no? Dahil meron lang tayong limited na oras, ifo-focus ko lang ang aking work at talk dito sa bacteria. No? Next slide, please. So ang bacteria, in comparison with the other 3 domains, ano, so usually sila ay simple lang. No? So baka sa libro, pag nagbabrowse kayo, makikita nyo, tuldok-tuldok lang sila. Either tuldok na pula, tuldok na blue, parang walang sense of architecture, no, sa kanilang body. But really, when you study them further, napaka-complicated pala on how they live their lives. Ano? They have characteristics that are very way different than other living organisms. No? Very unique, and some of them can even tolerate yung mga stress, no? At saka yung mga extreme environments. No? Next slide, please. So ngayon, sorry, meron din akong pre-test, ano? So ito, gusto kong itanong sa inyo, no? Why? Bakit nga ba natin kailangang aralin ang mga mikrobyo, no? So meron ako ditong 3 pamamaraan. Kung hindi nyo masascan ang QR code, pwede nyo i-click yung link, no? O kaya kung wala akong pang-click ng link o wala akong pang QR code, pwede nyo i-grab ang inyong keyboard at i-chat ang inyong sagot. Ano? So, meron tayong 1 minuto para hintayin ko ang inyong sagot. Bakit nga ba natin kailangang aralin ang mga mikrobyo? Can we do that? Why study bacteria? Okay, pwede po nyo i-scan yung QR code or use the chat box. So, ayun po yung link sa chat box natin para hindi na po nyo itatype isa-isa. Salamat, Janice. At kung kayo ay may camera sa inyong phone, pwede nyong i-picturean ang QR code na yan. O kaya, yung ating chat box. Yan. So, aabangan ko dito. Okay, ang ating kung may sumasagot na, ayan, no? May sumasagot na dito. Ayan, pumapasok ng ating mga sagot. Mukhang bibo ang ating mga guests this morning. Ha? Siguro, Jeff, can I borrow the screen for a while? And maybe I can share my screen para makita naman nila yung kanilang mga sinasagot. Yan.
Sure po, go for it.
Oo, pakisabi kung nakikita na nila yung screen ko. Ayan.
Yes, we can see it po.
Yan. Ayan po yung mga sagot nyo. So kung meron pang mga sumasagot, sige lang, no? I really wanted to highlight this particular answers. Ano? So sabi ng ating mga participants, no, we study bacteria to better understand health and disease. No, tama yan, no? For biotechnological applications, diversity, public health importance, to advance our idea on how life works, no, and in turn learn about how to make our lives better. So minsan tinatanong ko sa sarili ko pag nakikita ko tong mga sagot niyo kung high school ba talaga yung participants natin? Parang mga nasa grad students na itong mga to. Ang galing ng mga sagot nyo. Ayan, no? Important part of biodiversity. That's right, ano? So, minsan sinasabi nila yung mga mikrobyo daw ay bad. Pero actually, they are the small percentage of the bacterial world na bad. Karamihan sa kanila ay beneficial. Marami tayong mapapakinabang sa kanila.
Professor Len, for some reason po, yung screen na-cut off po.
Oo.
Maraming sagot, oh.
Reshare po siguro natin yung screen, Prof. Len, kasi ano po, truncated po, putol po.
Oo, ayan. So later, natin yan, ipaprocess ko yan. At gusto ko rin, meron din kasi dito sa chat box, gusto ko rin ikwento dito yung mga nasa chat box, yan, to know the benefits and bad effects of on our bodies and the environment, no, yan. Since they coexist with us in the ecosystem, no, kailangan nating aralin, ano. So, I think very good answers from everyone. I think nakuha nila yung gist, no, kung bakit natin aaralin. At kailangan aralin yung mga mikrobyo, no? So sabi nga nila, pag kasama mo, kailangan mong alamin, no? Sino siya? Paano ko ba ito pakikigsamahan? No? And those are very good answers. So thank you very much. I'll give you a copy of this one, Jeff, later so that we could highlight it in our events for today. No? Thank you very much for your answers. Sige, let's go back to my slide. Okay, so since sabi nyo, we need to study bacteria kasi kailangan natin silang makilala, no? Kailangan nating malaman ano ba yung role nila dito sa mundong ibabaw, no? Kailangan nating malaman kung cause ba sila ng sakit or hindi, or they are the ones to be utilized to cure a certain disease. no? So para natin sila maaral, ito yung mga various ways, no? So pwedeng traditional, pwedeng advanced, ano? So ito ang isang example ng traditional ways on how you can study bacteria. First, you can characterize their colony using their pure cultures, ano? So makikita nyo dito sa figure, alam nyo ba na yung mga kamay nyo, parang harmless naman, ano? Pero pag nilagay nyo yan sa culture media, ginagawa namin itong eksperimento sa aming klase, ganyan po yung lalabas. No? So kung nakikita nyo dyan, yan, no? Parang ang daming mikrobyo ng kamay nyo. So pag nakikipag-holding hands kayo sa inyong mga jowa, ano? Or kung saan-saan kayo hahawak, yan po yung mga nakukuha natin. Kaya tayo pinapaalalahanan na mag-hand washing routinely para yung mga hindi masyadong mababait na mikrobyo sa ating mga kamay ay mawala. Ano? And then, pag tumubo yan, you can actually isolate them isa-isa into pure culture. So, papaliwanag ko mamaya yung pure culture, ha? At aralin sila. Anong itsura, anong kulay, may amoy ba yan, etc. etc. No? So, in the laboratory, kailangan natin isipin anong media. Yung culture media, yun yung bago niyang tahanan. Galing sa kamay, pupunta siya sa media, paano ko siya papatubuin or paparamihin. Ano? So, yun yung talagang inaaral ng mga microbiologists. So, in colony characteristics and pure culture techniques, you really have to make sure na yung nakuha mo sa kamay mo, ang iyong aaralin at ililipat sa panibagong medium. At ang tawag doon ay aseptic techniques. No? So, inaaral po yan sa microbiology. So, next slide, please. Yan, sige. So, nabanggit ko kanina yung pure culture. No? So, bakit ba kailangan gawin ang pure culture? Ginagawa ito para malaman at aralin specifically ang isang uri ng microbial species. No? Halimbawa, siya ba yung salarin? Bakit nagkasakit yung animals? O siya ba yung salarin kung bakit nagkaroon ng water pollution? Siya ba yung dahilan kung bakit naging healthier yung isang pagkain and so forth, ano? So isa sa mga sikat na personalities dito sa pure culture ay si Robert Koch, no? Siya ang nagpauso ng Koch's postulates. Ang Koch's postulate ginagamit pa rin hanggang ngayon yan sa medical microbiology sa mga hospital. So kung kayo ay nagkasakit or nagka-experience kayo magkasakit, di ba? Sasabihin ng doktor sa inyo pag naka-admit kayo, i-culture po namin yung inyong dugo o yung inyong pupo o yung inyong urine or kahit na anong samples from you. Kasi titingnan namin kung merong mikrobyo at kung iyon ba ang dahilan ng iyong UTI o ng iyong diarrhea o ng iyong sakit. Ano? Because of Koch's postulate, they are able to identify the causative agent of a certain disease. No? At yon ang dahilan kung bakit a pure culture is very essential for us to study microorganisms. So, paano ba kinakarakterize ang isang pure culture colony? Next slide, please. Nung pandemic, no? Kasi pag hindi pandemic, madali lang. No? Nung pandemic, na-challenge kami lahat. I'm sure lahat ng mga nandito na mga estudyante ng college na merong microbiology, na-experience nyo siguro to na paano kami mag-microbiology lab, pandemic, di kami pwede magkita ni teacher, no? So, tinry namin ang virtual simulation, no? So, gamit ang kanilang mouse, computer mouse, no? Yun ang kanilang wire loop. At ikakalat nila yung mikrobyo para mag-produce ng pure culture. Siguro iniisip ng mga na-experience ng lab, ha? Paano yun? Hindi ba mahirap yun? No? Medyo mahirap po. Kaya talagang pinapalakpakan ko yung mga estudyante, no, na nagpakahirap dito sa virtual reality na ito na mag-produce ng pure cultures using virtual background, virtual websites. Ano? Next slide, please. Yan, dito sa software na ito, tuturuan kayo na mali ba yung ginawa mo? Sasabihin sa'yo, mali yata yung ginawa mo. Pwede mong ulitin. Yan, gano'n. Hanggang sa ma-perfect mo na, kagaya nitong picture sa gitna. No?
Ang
iyong
ganito, ma'am, sorry.
no?
Anong website po ito?
Pinakamahalaga dyan ay ang mapaghiwa-hiwalay mo into colonies ang iyong mixture. No? Next slide. Kaya lang hindi lahat ng estudyante namin ay may computer. Hindi rin lahat ng estudyante namin ay may internet. So ang pinagawa namin sa kanila, watercolor. No? So dito, paggamit ng aseptic technique sa paggawa ng pure culture, gamit ang watercolor at saka glitters. So ang idea dito ay maipakalat nila yung glitters na magkakahiwa-hiwalay. Yan, no? So at ipapaliwanag nila ano yung purpose ng tubig. Ano yung purpose ng paint or watercolor, ano yung purpose ng photo paper, ano yung purpose ng paintbrush, at ano yung purpose ng glitter. No? I'll be sharing later on the website for the virtual lab para ma-share natin sa ating mga teachers na nandito sa room para maipatry niya rin sa mga estudyante niya. No? Sige, next slide please. Hindi, yung previous slide. Yan, okay. So in the real sense of the laboratory setup, ganito yung itsura ng mga colony na makikita nyo after pure culture. And there are four ways wherein you can characterize your pure culture colony. Number one, ano ang kulay or what is the color? Because they can vary their color into cream, white, yellowish, no? Meron ding pinkish, may orange, may yellow, and so forth and so on. So that's the first one. The second one is form, no? So merong mga iba't ibang form dyan. Meron kami actually kodigo, mga microbiologists, ano nga bang itsura? At titingnan namin yung kodigo na yan. Sila ba ay punctiform, parang tuldok-tuldok na maliliit, circular, filamentous, irregular, no? Hyoid or spindle, no? So titingnan mo lang dyan, no? Ano bang itsura ng aking colony doon sa iyong kodigo. The third one is elevation. Gano'n sila kataas. So minsan titignan mo yung iyong petri dish pag ganun, no? Ay, gano'n ba sila kataas? Sa halip na paharap sa'yo. And then you can see whether it's flat, no? Or raised. Kulang-kulang yata yung spelling ito, no? Flat or raised, no? Or convex or fulminate, umbonate, etc. No? And then also the margin, yung kanyang mga edges, no? Titignan mo. Sige nga, tingnan natin. Paano natin i-characterize ito? Ating colony dito sa picture. What's the color? Can you try? Ay, sandali lang po. Yung previous muna. Yan. What's the color? Can you type in the chat box? No? And then what's the form? What's the elevation? And then what's the margin? Sige nga. Tignan natin. Try lang natin. Don't worry, walang minus. Walang minus kung mali. Let's see that. Yan, may mga nagtatry na ba tayo? Ayan. So, apat color, form, elevation, and margin. Let's see. Sinong maglalakas loob na mag-try? Color daw ay greenish. Okay, so you can actually just simply state that color, greenish colony, no? Or green colony, or light yellow colony. Yan. So may nagsabi na circular, convex, entire. Green colonies. Tamo, talaga tingin ko, hindi talaga high school, Sir Jeff, itong mga na-invite mo. Magagaling ito. So this particular colony is a light yellow. No? So kung iba po yung kulay sa screen nyo, okay lang yan. Mapapatawad po kayo ng screen. So light yellow, no? Colonies, no? Circular. Okay. And these are convex colonies, no? And with entire margin. Ayan, oh, very good. Palakpakan nyo naman yung mga sumagot sa atin sa chat box. Oh. Ang gagaling naman po talaga ng ating mga estudyante this morning. Thank you for answering that question. Oh, meron akong kasunod. Mas challenging ang itsura niya. Next slide, Sir Jeff. Tingnan natin kung kaya nila itong second nating slide. O, sige nga. Color, form, elevation, and margin. Try. Medyo bongga po yung kanyang itsura, no?
Ang ganda naman niya.
Ang masasabi ko lang po dito ay ang masasabi ko lang po dito ay Sus Mariosep. Ano yan?
Oh, kanina nga napapaisip ako, sabi ko kaya pala hindi ako biologist.
Apo, and just to give you an idea no.
These are bluish green kung medyo gusto nyo ng specific fluorescent, no? Fluorescent green. No?
Anong bacteria po ito, ma'am?
So ito na yung hyoid eh. Ito na yung ano. Okay. So ito na yung gumagapang. Yan. Okay. At ito na yung tinasawag natin na irregular ang kanyang margin of filamentous yung kanyang margin. Very good. Ang galing-galing naman. Sorry po, parang mali-mali yung spelling dito. But anyway, pag kayo po ay microbiologist, minsan may mga kodigo naman yan sa inyong laboratory para kayo ay magtingin, no, na pag-characterize. Basta ang mahalaga, tatandaan nyo para makaracterize ang colony, color, form, elevation, and margin. Kaya lang, hindi lang po tayo sa petri dish mag-grow ng ating mga mikrobyo. Minsan, kailangan natin silang i-grow sa test tubes. No? So, next slide, please. Pwede rin silang i-characterize pag sila ay nasa test tube. Ibig sabihin, ano bang itsura nila nung tumubo na sila sa solid slants, yung mga nakaslanted na test tube, or sa broth culture o yung sa liquid medium. No? So, meron din tayong tinatawag na characterizations kapag sila ay nasa test tubes. Ang mga ito, no, ay tinatawag natin na colony characterization. Napakahalaga itong start para ma-identify nyo or at least maihiwalay mo na siya doon sa mga iba't ibang mikrobyo at first base ka na sa identification. No? Next slide, please. Ang sunod ay ang microscopy. Okay? Siguro isipin nyo, meron bang option sa colony? No? Meron bang advance niyan? Ano? Wala pong advance, talagang talent ng microbiologist ang pag-aral at pag-describe ng colonies. Pero ngayon, dito sa microscopy and morphological characteristics, gagamit tayo ng traditional at gagamit tayo ng advance. Nasa inyo yun kung ano ang kailangan. So, ang microscopy ay important sa microbiology kasi po, hindi natin sila nakikita sa naked eye. Yung colony na pinakita ko sa inyo kanina, parang, okay, isang colony. Pero yung isang colony na yon, pwedeng maging hundreds or thousands, 100,000s na cells in one colony. So, imagine niyo, ang dami palang laman nun. Ano? So, kailangan nating aralin in addition to the color, the form, the margin, and elevation, anong itsura nila? Anong shape? Anong size? No? Meron ba silang extra structures? No? Sila ba ay cute or nakakatak-cute? No? Kaya kailangan natin ang microscope. Ano? Next slide, please. I think high school, ini-introduce sa atin ang parts of the microscopes. Ano? So, dito sa microscope, tinuturuan tayong gumawa ng wet mount. No? So, kukuha tayo. Ang alam ko, nung high school ako, pinagawa kami ng hay infusion. Sino nakaka-relate? Reacts nga dyan or taas ang kamay. Ano? At maghahanap tayo ng mga gumagalaw-galaw na creatures mula sa hay infusion gamit yung ating microscope, ano? So kami nung tuwan-tuwa, wow, nagsuswimming, ano? Tapos sabi ng teacher namin, okay, start to identify. Ay, ma'am, wala po yung gumagalaw. Talaga yung mga estudyante, magaling magpalusot, ano? Pero ito yung beauty ng microscopes. Maybe, pagtingin mo sa hay infusion, ay, nabubulok na damo-damo lang yan. But inside the microscope, you can see the beautiful, beautiful world of microorganisms, ano? Next slide, please. At ibigay ko sa inyo yung traditional ways and traditional approaches on how to characterize them morphologically using microscope. Approach number one, simple staining. So pag sinabing simple staining, gagamit lang kayo ng isang uri ng dye or coloring agent. Bahala ka kung anong meron sa inyong eskwelahan or kung anong gusto mong kulay, blue, red, green, yellow, bahala ka, no? Ang mahalaga nito, makikita mo na kaagad ano ang kanilang shape and you can measure their particular size. So very simple, you just have to prepare your smear. In preparation of your smear, you have to pass it sa heat, no, gamit ang inyong Bunsen burner or yung alcohol lamp. In this way, mamamatay na po yung cells natin sa ating staining method. Unlike kanina doon sa hay infusion nating wet mount, lumalangoy-langoy pa sila, no? Pero dito, we cannot afford studying their shape and their structures habang lumalangoy-langoy sila. So kailangan natin silang i-fix, no? Kumbaga parang pumirmi ka dyan, no? Parang gano'n, no? And then when you fix them, you can apply the stain. Just one stain, that is called simple staining. And under the microscope, using your OIO or yung oil immersion objective, dahil po sobrang liit nila, we need a high power objective. Ganyan yung makikita nyo. So you can describe them, bilog ba sila? Bacillus, rod, spirillum, etc., etc., and what will be their sizes, no? Next slide, please. Second approach, differential staining. No? So, hindi pa ako masaya na nakita ko yung kanilang shape or nasukat ko sila. Gusto ko kasing malaman ano yung kanyang mga iba pang information. One important information is yung tinatawag na differences in their cell wall. Okay. Narinig nyo na ba kung sino or kakilala nyo na ba? Ayan, si Christian Gram. Christian Gram po kaya mag-iba ng spelling, no? Christian Gram. Baka kung ano-anong kung saan tayo makarating, no? Si Christian Gram ang nagpauso nitong very, very important, no, na staining method. Correct. Ayan, may bibo na dito sa chat group. Ang gram staining. Ayan, sabi ko nga sa mga estudyante ko po sa microbio, kalimutan nyo na lahat sa microbio. Wag na wag ang gram staining. Why? Because this is a very valuable differential method, no, to differentiate the cell walls of bacteria. Okay? At na-divide na ang inyong bacterial world into two. Gram positive, nag-stain siya, okay? And gram negative, hindi siya na-stain ng iyong primary color dye, but instead, ang kinapture niyang color is the second dye. This is very important because this will tell you ano ang cell wall characteristics ng iyong bacteria, no? So very good sa mga nasa chat box. Thank you for answering, no? Gram staining is discovered and of course shared to us by Christian Gram, no? Next slide please. So kung ang simple staining, may isa tayong stain, differential staining, may dalawa na tayong stain, no? Para makita mo yung difference. Dito sa ating mga microscopes na pwedeng walang masyadong stain, no? So sabi kasi nila, no, pwede bang dark field lang, no? Walang masyadong stain, no? So this is a yeast, no? I chose a very nice picture of a yeast, not a bacteria, no? But you can also use this for bacteria, of course. This is important to let you know and contrast yung pag ina-upgrade mo yung microscope mo, na-upgrade din yung view mo, no? So if you wish to check out for other structures, you can make use or upgrade your microscope. Kasi tignan nyo, no? Pag sa bright field microscope, okay, I can see the shape, I can see kung paano sila naka-arrange. Pag gumamit ka na ng phase contrast, medyo iba na siya, no? At pag gumamit ka ng dark field, ma-emphasize ang kanyang mga special structures. So it really depends on ano bang kailangan ko. But usually, if you wish to just characterize the structure of your organism like this one, gusto ko lang makita naman yung shape and arrangement, a bright field microscope is already enough. Next slide, please. Ayan. Mikrobyo po yan. Nanggaling po yan sa river. Ilog. Kumuha sila ng sample. Okay. Tapos, nilagyan nila ng tinatawag na probe or dye. No? So, may marker. No? And then, bawat marker na yon ay magpapakita ng iba't ibang kulay ng mikrobyo. Halimbawa, bigyan kayo ng analogy. O lahat ng magaganda ay magiging kulay green. Example, gano'n, no? So, lahat ng mga pogi magiging kulay blue. Lahat ng mga hindi pinagpala magiging kulay violet, no? So pag lahat tayo ay nagsama-sama because of our marker, para meron tayong tag, no? Kung maganda ka, hindi pinagpala or pogi ka, no? So pag tinignan tayo under the microscope, ganyan po yung magiging itsura natin. So this is an advanced way for us to characterize morphologically microorganisms, no? So kita nyo, iba't iba na yung kulay sa kanila, and you can design your probe or your dye, no? Ano bang dye ang gusto kong ikabit, no? So this is now advanced, yung mga advanced research, gumagamit sila nito para malaman kung sino ba, no, sa mga microorganisms na yon ang may certain na characteristics na kanilang binabantayan, no? Next slide, please. So another approach, medyo advanced na rin to. Why not make it 3D? Three-dimensional. Ang kita nyo kanina, two-dimensional lang, di ba? Okay, anong shape, anong measure? Bright field microscope. But when you use a 3D microscope, no, you can, parang nakalutang sila, no? Nakapop up sila. No? Kagaya nitong dog water bowl. Kumuha sila ng samples dun sa painuman ng kanilang dog, no? At using the atomic force microscopy, they saw this wonderful, beautiful specimen, no? So yung mga high-end na microscope gaya nitong 3D, gagamitin lang yan kung kailangan mo ma-view ang special structures. Kung hindi mo naman kailangan i-view ang special structure, hindi mo kailangan bumili ng atomic force microscopy, okay? Or microscope. Next slide, please. Okay. So, hindi ka pa masaya sa 3D, we can use laser. Ayan. So, yun yung tinatawag nating confocal laser microscope. So, tingnan nyo dito, no? So, kahit sama-sama yung mga mikrobyo, no? Madi-differentiate nyo sila using confocal laser microscope, ano? So, gumagamit din ito ng mga tracking na component or molecules or dye, no? Para ma-identify mo sila between or makategorize mo sila between the different mixture of microorganisms. Okay? Next slide, please. After laser, why not electromagnet? No? So, ito yung mga electron microscope. No? So, you have your scanning. Pag gusto mong makita yung buo, pinalaki mo lang yung view, no? Using from the bright field microscope. Or the transmission electron microscope. Kung gusto mong makita naman yung loob. No? So, kita nyo dito, no? So, you don't really need to buy such microscopes, pero meron tayong mga institutions na meron ito, no? Kagaya nga nung UPLB Biotech was so helpful with us noon. Wala kasi kami noon na electron microscope. So tinulungan nila kami to view using scanning electron microscopy whether dumidikit ba yung mga heavy metals sa surface ng aming mikrobyo. Yan. So this is very nice if you would like to venture on the use of this one. This is really very nice, no? And then also, meron tayong mga microorganisms like the viruses na kailangan talaga electron microscope kasi very, very small na sila. No? Next slide, please. Okay. So, kanina, colony, characterization. Next, nalaman nyo na rin paano sila i-characterize using the microscope for their morphology. The next phase is physiology or paano ba sila nagme-metabolize para mabuhay. So bakit ko kailangan aralin yan? Kasi ang iba't ibang mikrobyo ay may iba't ibang ways, no? Iba't ibang ways kung paano mag-survive, kung paano kumain ng mga nutrients and so forth and so on. And this can be shown sa inyong mga tests, no? There's a long array or battery of tests, ano? At minsan ang ganda-ganda nilang tignan, no? Very laborious kasi I'm sure makaka-relate ang ibang college students in the room right now na it will take about 1 to 2 days for them to complete lahat ng mga culture media for the physiological test. I-autoclave pa nila yan doon, tapos i-inoculate, and then kinabukasan pa nila yon malalaman yung resulta. No? But all of these particular information will give you an idea what is the identity of your bacterial strain. And there are some references like this one in the slide, ano, na meron na silang algorithm na sinusunod. Ano? So halimbawa, ginawa mo itong test na to, meron plus, minus, sino nag-positive, sino nag-negative, until eventually, it will lead you to the right species name or the right species identity. No? Kaya mahalaga ang gram staining. Kasi alam mo na agad, ang gagamitin ko ba ay test, physiological test para sa gram negative. So may flow chart na yan. At para sa gram positive, may flow chart na yan. So it cuts your work in half. No? That's why napakahalaga ng gram staining. Isang halimbawa ng physio test ay ang catalase test. Ano? Ang catalase test ay pag-check whether ang mikrobyo ay may enzyme na tinatawag na catalase. No? Ito yung kapag na-expose ang isang mikrobyo sa hydrogen peroxide, yung nilalagay nyo po sa inyong mga sugat para luminis, ma-disinfect. Ang mga mikrobyo minsan meron silang enzyme para labanan ng effects ng hydrogen peroxide. No? So, hindi po lahat meron nun, so mahihiwalay mo na kaagad ang iyong mga mikrobyo sa pamamagitan ng catalase test. So, meron pa akong ilang halimbawa sa mga susunod na slides. Next slides, Deb. Halimbawa, ang MRVP test, no? Ito yung nagbibigay sa atin ng idea whether pag binigyan mo ba ng sugars or glucose ang iyong bacteria, anong ipoproduce niya? Acid, alkaline product, wala siyang pake. O kaya, hindi ako kumakain ng sugar. No? Meron pong ganung mikrobyo. No? And because of that categorization and differentiation, mahihiwalay mo na siya and you can identify them later on. No? Next slide, please. Pwede rin tayong gumamit ng gelatin hydrolysis test. May mga mikrobyo na may enzyme called gelatinase, kaya niyang i-liquefy yung ating gelatin. No? Next slide, please. Or lipase. Sabi nga nila, meron mga mikrobyo na may enzyme, tutunawin niya yung taba. O kaya, i-utilize niya yung mga fats and lipids to produce their valuable products na kailangan nila sa metabolism. So again, hindi po lahat ng mikrobyo ay may ganyan. So very important na gamitan natin ng mga physiological tests to identify them. No? Next slide, please. And then beta-lactamase test. No? So mga traditional to, ibig sabihin, ang dami nyo pong gagawin ng culture media before kayo makapaggawa ng physiological test. So isa itong beta-lactamase test para malaman nyo kung meron ba siyang enzyme, no, na nagbibigay ng capability dun sa mikrobyo para ma-resist niya yung action ng mga beta-lactam antibiotics. So may chance na pwede siyang maging resistant sa beta-lactam antibiotics. No? And because of this test, no, meron pang mga susunod na test para malaman mo kung marami ba ang antibiotics ang kaya niyang i-resist. Napakagandang example po ito or idea or information para sa mga doctors para pag nagkasakit kayo, tas ito yung mikrobyo ng profile na meron siya, iiwasan niya yung mga antibiotics na resistant yung mikrobyo na nakuha sa iyo. No? So these are the traditional ways. Ano? And sabi nga nila minsan nakakapagod yung traditional ways. That's why there are some groups years ago na nakaisip na bakit hindi natin gawing madalian yan. No? Pwede bang mapadali yan? Yung kukonti na lang ang gagawin kong test tubes, culture plate, flash na media. No? So, meron tayong tinatawag na advances. No? Next slide, please. Ayan. Nandiyan na po lahat yung ginawa yung culture media. Sa kit na yan, ang bawat isang, ano bang tawag dyan, cylinder ba? Anong shape yan? Oval na yan, ay may culture media. Ilalagay nyo na lang yung inyong mikrobyo, whole culture suspension, wait 24 hours, at sasabihin niya sa'yo, yan ay E. coli. O diba, ang saya-saya ng buhay. No? So, yun yung advanced way. Hindi ka na mag-autoclave. Hindi ka na mag-mimix ng media, and so forth. No? So, these kits are available in the market, and if you have the budget, you can actually utilize it. This principle revolves also based on the physiological characteristics of your microorganisms. Kasi bawat isa pong bilog na yan ay merong raw materials for them to metabolize. Ano? May sugars, no? Merong mga nitrogen compounds, etc., etc. At ngayon, marami na silang category of kits, whether kayo ay gram-negative enteric, gram-negative non-enteric, ikaw ba ay gram-positive, ikaw ba ay yeast, ikaw ba ay lactic acid bacteria. Kaya again, important, number 1 step, para ka makapag-identify, gram staining. Okay? Sige, next slide please. So another advanced way for you to profile yung mga physiological, ayoko kasing maglagay ng mga ganun, no, na isang kit lang. E paano kung yung buong soil sample ko, ang gusto ko nang aralin? Kasi yung kung slide kanina, isa-isang colony. This more advanced platform called the Balolong EcoPlate, you are going to profile the physiology of the community ng mikrobyo sa sample. Pero nyo, gagawa lang po kayo ng soil solution or sample solution, and then you will put it in the plates already. At bibigyan na kayo ng idea ano yung profile ng physiology nung soil na yon because of the microorganisms. This will also give you an idea anong ginagawa nila sa aking lupa. No, parang ganon. So if the microorganisms will help you enrich the soil for agriculture, or if this particular microorganisms, kunwari galing silang mining soil, ay may enzymes to degrade the heavy metals, aba, magandang application po yun. So, napakaganda ngayon, ano, itong advances na ito. Kasi, again, hindi na po kayo magagawa ng culture media. Hindi na rin po kayo magagawa ng pure culture. Just your sample. Ida-drop na lang doon sa mga wells ng iyong 96 well plates. No? So, thanks for advances. Nakakat ang ating mga trabaho in half. No? Next slide, please. And now, syempre, ang pinaka-paborito nating advances ay ang pagkakaroon ng PCR o ang polymerase chain reaction. Dahil po sa PCR, we have the power now to study our genes. No? Biruin nyo, dati ang inaaral nating gene, putol-putol. Alala ko, nung college kami, yun ang inaangin na tinuturo sa amin. Segments ng genes, no? Putol-putol. Random. No? Pero ngayon, pwede na nating aralin ng isang mahabang gene segment, isang buong genome, or the whole gene of the community. So imagine that. And of course, we would like to thank Karen Mullis for this very important breakthrough for science. Nanalo po siya dyan ng Nobel Prize. No? So tingnan natin anong pwedeng gawin kapag alam mo ang PCR. Next slide, please. Ayan. So, pwede po tayong mag-meta-genomic analysis. No? So, ito yung combination ng pag-aaral natin ng genomics, meaning, alam mo kung sino yung mga nandyan, taxa, species name. No? Kanina, ang dami natin pinagdaanan, no? I-describe ko yung colony, magma-microscope ako, mag-physiological ako, and then, yehe, meron akong tentative identification. But for genomics here, no, bibigyan ka na niya based on a database, no, ng pangalan. Ganun na po ngayon kabilis, no? So the information is so robust, no, that even if the gene segment na nakuha sa sample mo ay wala pang pangalan, i-report din yan, ano, na uncultured operational taxonomic unit or OTU, no? So also, in addition to that, sasabihin din na niya sa'yo ang function ng mga microbiome yon doon sa iyong sample. So imagine the possibilities of samples: soil, water, food, air, meron pa kami dating sample na dust, ano? And even your own tissues. Fecal sample, saliva sample, ano pa maisip nyo? Sweat, blood, breast milk, skin, ano pa, no? Pwede nating gamitin, ano? So this is a breakthrough on genomics that we can study the microbial profile and of course the function of microbial community na po ito, ha? Hindi na isa-isa, no? Microbial community using metagenomics, na? Next slide, please. So sa metagenomics, ang sinasabi nila, ito yung dry lab. Hindi ko alam kung familiar na kayo, no? Paki-reacts nga. Narinig nyo na ba yung dry lab at saka wet lab? Sino nakarinig na? Heart reacts or thumbs up naman dyan, no? Sabi nila yung wet lab, hindi po na parang umuulan yung laboratory nyo dahil sira yung pipe, ha? Yung wet lab, yun po yung dinescribe ko kanina, no? Magagawa pa kayo ng culture media, gugrow nyo yung mga mikrobyo, no? Pero yung dry lab, nakaharap lang kayo sa computer. You are analyzing such data. I think I have some students here, nakita ko kanina na nagda-dry lab ngayon. Yan, pa-heart naman kayo dyan. So ang dry lab utilizes bioinformatics data, database, no, information. Lahat ito ay nanggagaling sa computer. Gagamit kayo ng mga software sa computer, okay? At magbibigay kayo at mag-generate ng new information using the data. Kaya dry lab pa rin siya. Nag-experiment ka pa rin, nagagawa ka pa rin ng hypothesis, nagbibigay ka pa rin ng new questions, kagaya ng wet lab, ano? So here are some examples of some softwares that we usually use, yung mga sequence alignment like your Clustal, or yung mga data analysis, no, kagaya ng Silva NGS, no, to make your corona plots, no. Next slide, please. Yan, sige. So kanina may nabanggit akong metagenomics, no. So may dalawa tayong pamamaraan ng sequencing. Yan. Kasi kanina, may nabanggit siyang next generation sequencing. Dalawa po yung pwedeng paraan. Yung una is amplicon-based. Pag sinabing amplicon-based, ang amplicon ay produkto ng PCR. Ibig sabihin, maigsi lang siyang segment. Example, 16S rRNA gene. Bitin ko, ha, 16S rRNA gene. Ito po ay isang bahagi lang, no, ng DNA ng iyong mikrobyo. Pero ang power nito, no, kaya meron dyan ng mga V1, V3, V3, V4, yan ay mga segments na napag-differentiate niya ang lahat ng mikrobyo so that your software can specifically identify and differentiate that into a species, a genus, a class, an order, and so forth and so on. Amplicon-based using 16S. Pag mikrobyo po yon na prokaryotic, ibig sabihin bacteria at saka archaea. Pag eukaryotic, pwede kang gumamit ng ibang gene kagaya ng 18S. No? So, isang example po yan. May isa pa akong pa-arrow dito sa baba, yung shotgun. Ano yung shotgun? Napaka-violent naman yata ng shotgun, ano? Yung shotgun, ibig sabihin po nun, hindi lang bacteria ang gusto mong makita sa iyong sample. Kunyari, tubig. Hindi naman bacteria lang nandun, ano? Dugo, pupo, soil. Hindi lang bacteria. So, using your shotgun metagenomics approach, lahat makikita mo doon. bacteria, fungi, virus particles, archaea, and so forth and so on. Makikita mo doon. So ikaw, anong gusto mong aralin? Gusto mo ba specific lang? So you can try the 16S based approach or the amplicon approach or shotgun. Carry lahat. No? Lahat pwedeng aralin. No? Sige, next slide please. At after nun, ang kwento ko po sa inyo ay genomics pa lang. Ano? Marami nang sumunod na pinauso, ang tinatawag nilang multi-omics. Kasi minsan, ang mga tanong natin ay sunod-sunod. Ganun sa science, di ba? Pagkatapos mo na isang experiment, nasagot mo yung hypothesis, may panibago ka na namang research question. No? So pag-aaral ng genomics, it will answer the question, what can happen? Ano kayang pwedeng mangyari? No? So the genes, the gene information, ano mga genes na nandun, yun ang magsasabi sa'yo na since may gene siya for resistance, pwede siyang maging resistant. Transcriptomics will tell you what appears to be happening. Ibig sabihin, okay, so may gene siya for resistant, naka-turn on ba? Magaganap kaya yon? Okay, so pag naka-turn on because of your transcriptomic data, then you are very sure that pwede siyang maging resistant. Next analysis, proteomics. Ang tanong sa proteomics, what makes it happen? Teka, paano siya na-turn on? No? Proteomics will give you a profile of proteins to make resistance possible. No? So ito po yung mga proteins niya na nasa cytoplasm. Ito yung mga proteins na nasa outside, yung nasa cell membrane. No? And then, ito po mga proteins na to, tumutulong para ma-turn on ang resistant or resistance genes. So that's proteomics. And then metabolomics will give you the answer to the question, what has happened? Kaya pala siya naging resistant. Na-produce nga niya yung protein of interest. Nagets niyo po ba? So pabalik, when you have data for metabolomics, andun ng protein, that's why resistance happened. Okay? Sa proteomics, itong protein na to, the reason why it's resistant can be found in the cytoplasm. And this protein in the cytoplasm can be turned on by this certain gene. At itong gene na to ay matatagpuan sa this part of the whole genome. Okay? Nasundan niyo po ba yung kwento? Ang tawag doon ay multi-omics approach. Hindi lang antibiotic resistance ang pwedeng aralin dyan, pwede niyo ring ipaliwanag. Bakit may diabetes? Bakit may obesity? Bakit nagkakaroon ng cardiac problems? And so forth and so on. Pwedeng mikrobyo, pwedeng pantao, pwedeng halaman. That's multi-omics approach, and it's a highly valuable advanced way to study microorganisms, even nasa community siya. No? Sige, next slide please. Okay, meron ulit akong tanong. Yan, meron ulit akong tanong ha. So grab your keyboards again, at gusto kong itanong sa inyo, which one is more important? Traditional or advanced? Yan. Sige nga. Tingnan natin. Nakabangan ko din itong sagot nyo dito. Sarap ko yung aking... Yan. You can try, or you can again do the keyboard. Ako nga po yung mga sumasagot na dito. Yan, yung mga mangilan-ngilan sumasagot na. You can use the chat box, ha. Sabi po nila, oy, marami-rami yung kumakampi sa both are important. Both are important daw. Pero sabi ng iba, I think because of the traditional way, it leads to the advances. Very good, Iggy, no? Ang ganda ng insight mo, no? Kasi oo nga, no? Because of the traditional ways, we have pinpointed and realized some gaps, no? Na paano kaya natin ito masusolusyonan? there came your advances. Wow, very good, Evie. No? So ayan, so may nagsagot na both are important, advances are important, traditional are important. Very good. And then sabi ni Liriel, traditional because we cannot develop unless we utilize the traditional as a reference to improve. Very good. Ano? Salamat sa lahat ng sumagot. Nakakaliw, ang daming sumasagot. Sa tingin ko talaga, the future is bright for Philippine STEM. I think you are in good hands. Salamat po sa mga teacher na nag-encourage sa inyo sa science, technology. Maraming salamat po. Let's go to the next slide para malaman nyo kung bakit nga ba important ang traditional and advanced together. Okay? So, tingnan nyo po to. No? So, bibigyan ko kayo ng halimbawa. Kung gusto nyong aralin ang E. coli, aralin natin yung colony, di ba? O anong itsura ng colony? Anong itsura niya sa microscope? Anong itsura niya sa PCR? Anong itsura niya sa physiological, whether traditional or advanced using the kit? That's E. coli. Pero pwede niya ring aralin ang E. coli. Nilagay ko yung arrow dyan ng E. coli. No? Sabi ng lahat ng mikrobyo sa community niya sa soil. And that's the advanced way. That is metagenomics. So in metagenomics, it can be coupled with your traditional way. Yan po yung tinatawag namin ngayon na culture-dependent at saka culture-independent approach. Kasi mahalaga na maaral pa rin natin yung mga mikrobyo na nakita natin sa metagenomics. Anong itsura niya in culture? Mabubuhay ba siya sa culture? Hindi. Meron ba siyang pinoproduce na metabolites na very important for us? Kaya dun sa sumagot ng both are important, salamat. No? At dun sumagot din na I think traditional is okay or advanced is okay, thank you very much then. Tama po kayo lahat. Ano? Both are really important. So kailangan nating pag-aralan ang traditional at saka ang advanced ways so that we could really maximize studying bacteria. Okay, next slide, please. And because of those things, I would like to introduce to you, I think this will be the last set of my slides. You know? Our small laboratory, maliit lang po kaming grupo sa University of the Philippines, Manila. We call ourselves Applied Microbiology for Health and Environment Research Group or AMHERG. Dumarami na po kami ngayon, kaya yung ibang pictures, wala pa dyan. Ano? Next slide, please. Ikukwento ko sa inyo kung ano yung mga ginagawa namin. First is COVRAS. Kung naririnig nyo na po yung wastewater-based epidemiology, meaning yung po mga wastewater natin, yung mga kanal tubig ba? Ganyan. Parang mga taong kanal kami, no? Yung mga taong kanal, no? Kumukuha po kami, kita nyo yung mga picture ng mga RAs ko na napapastop kami ng traffic, no? Sandali lang po, kukuha lang po kami ng tubig dito sa imbornal, ano, or sa manhole. And then we utilize that. This is our project. This is funded by the DOST, PCHRD, and a very generous NGO, Care Netherlands, ano, so that we would be using the samples to monitor, no, and devise a way of predicting your risk of COVID, no, and other waterborne diseases, as well as antibiotic resistance using that one. So kung kayo po ay tinetext ni NDRRMC ba yun na may baha sa inyong lugar o kaya may lindol sa inyong lugar, susunod na po ito. No? Ang inyong lugar ay at risk sa COVID-19. Maaaring iwasan ang bahaging ito ng inyong water system. No? So pwedeng ito ay ilog, ito ay inyong lugar, baka gusto nyo magpa-test. Yan. So sa halip na iintayin pa namin kayong magkasakit, this is our very good way to contribute prevention of COVID-19. No? So sila po ay COVRAS. Yan. Next slide, please. Meron din po kamang project na tinatawag na Cortex Sales. Ito naman po ang aming contribution para sa maritime industry. Ang ating pong maritime industry under the International Maritime Organization ay kailangan mag-comply sa standards na ang kanilang ballast water. Meron pong tubig kasi sa tangke.
Kain ako pag natapon yung pagkain ko.
Magdadak. kailangan mag-re-release sila ng tubig. Magkukuha ng tubig, no? At kung sila ay aalis, magbabawas naman sila ng tubig. So we have to make sure that the water they draw or the water they release are free from invasive organisms as well as antibiotic-resistant microorganisms for compliance. No? Next slide, please. Meron din po kaming conserve kaigangan. Meron ba tayong mga taga-Samar dito? Kaway-kaway naman tayo. Meron po kaming partnership with Samar State University funded by Picard. Ito po yung inalam namin, yung mga mikrobyo, no, na nasa limestone forest. Ito yung mga talagang parang bato-batuhan ito, na bundok, ano? At kumuha kami ng mga rhizosphere soil and have a look, no? At ano yung mga mikrobyo doon, no? And we have very, very nice, important information, which is very important for public health and tourism, no? So abangan niyo po yung aming mga reports soon. No? Next slide, please. And then another important project for COVID is this assessment of gut microbiome for coronavirus disease or COVID-19. Ngayon po, ang mga test natin na ginagamit ay oropharyngeal, no, nasopharyngeal testing, saliva. So ito pong sa amin, baka pwede rin gamitin yung inyong mga pupo. No? So yung mga pupo nyo ay pwede na rin gamitin para sa testing. Yun po yung essence ng project na ito. So ongoing po ito. So excited kami malaman na pwede nga ba ang inyong mga pupo. So sa next time, sa halip na ayaw nyo kasing pasuksok yung ilong nyo, no? Pag sa testing, baka pwede magsasabit na lang ako ng pupo sample. No? Next slide, please. And then the Field Diamond Project. So this particular project is funded by PCHRD, a 3-year project. Ito naman po ay tungkol sa lactic acid bacteria, yung aking lab life. Yan. So ang gusto namin dito ay makadevelop ng lab-based biofunctional food para sa ating mga HIV patients na nag-undergo ng antiretroviral therapy. So kami po ngayon ay nasa year 2 na. We are now developing that product. And hopefully, no, at the 3rd year, we are able to explain the mechanisms of this one. No? Next slide, please. Ito naman sa hindi kailangan ng laboratory, tumutulong po kami para malaman yung mga issues related to pandemic zoonosis. Kapartner po namin dito ang York University in Canada. Next slide, please. At nakikipagtulungan din po kami sa Roche through the Project ECHO. Ito naman po ay tungkol sa prevention or early detection of cancer patients para po ito ay training, research para naman sa ating mga barangay health workers. Next slide, please. Okay. So, yun po yung mga kaganapan namin sa laboratory. Gusto lang namin kayong i-inspire kahit papano, no? And be one of us, no? At Unheard City Manila, you can email us if you're interested, no? Gusto namin kayong i-encourage na pag-isipan. San ka ba magaling? Ano bang mga bagay na kapag ginagawa mo yon, na-appreciate ka, no? At syempre, ang pinakamahalaga, tignan nyo, yun bang ginagawa mo makes people lives better. Kapag nag-intersect po yan, that is your calling. And for us at Unheard, our calling is to utilize microbiology, now for health and environment. Of course, for the betterment of the Filipino people. No? So sabi nga ng damit ko ulit, lagit-lagi para sa bayan, we use microbiology to help the Philippines move forward. So I think this is my last slide. Thank you very much, and I hope you have questions so that we could chat around a little more. Thank you very much.
Ayan, ako maraming salamat po, Professor Len. So, we will give Professor Len a few minutes to drink water and to refresh naman kasi solid, more than an hour of talk. And then after that po, we will proceed to the question and answer to the open forum, no? So we encourage all of our students to send us your questions, no? So there's a Zoom chat box, sa YouTube live chat box, and then kung meron ditong matapang, kung merong matapang na audience members, feel free to raise your Zoom hands, and then we'll acknowledge you and will allow you to ask your questions live. Ayan. So Professor Len, feel free po if you need to drink water, mag-ano na muna po, magpa-plug din po kami ng upcoming events. So, I will be sharing a few slides with you for our like to share with you some of our upcoming events. So, as part of the Filipino Science Hub Biology Month, yung first installment nga natin ay yung kay Professor Len. So we have 3 more after this. So on April 9th, we will have Dr. Mark Calabon, and he'll be talking about aquatic mycology. So that's going to be April 9th at same time. So Saturday at 10 in the morning in the Philippines. And then after that, magkakaroon tayo ng Holy Week break. So wala tayong session on April 16th. The third installment will feature Dr. Glorian Nike Lazaro. So she'll talk about and nuances of cancer drug discovery. So kung kay Professor Len ay microbial discovery, ito naman cancer drug discovery naman with na medyo bio yung approach talaga. And then we will conclude yung Biology Month webinar series with Professor Eufrosina Atabay, and she's from CLSU, and she'll talk about the transformation of the Philippine carabao. So I'm hoping I'm not butchering the pronunciation of the scientific name. So Bubalus lean. So from a beast of burden to a beast of fortune. So please watch out for those events. I know that most of you have registered as mga events na to kasi I was monitoring yung Zoom statistics. So watch out for all of these. And then siguro I would also share another set of slides just to siguro share with our high school community the myriad of materials that we have released over the course of 2 years in support of the STEM community. So sa Phil-Sci Hub. So most of you, you're pursuing, you're actually nasa STEM strand yata kayo sa, for example, sa Bataan National High School. And most of you, merong mga capstone projects, So, kailangan nyo mag-perform ng scientific research in high school. So, we would also like to share that Filipino Science Hub has actually developed a portfolio of training courses for research. So, essentially from ideation, review of literature, proposal writing, experimentation, data processing, hanggang scientific writing and conference presentation. We have that portfolio of training materials in Taglish. So essentially, kung paano yung pagkaka-deliver ni Professor Len na parang nagchichika-chikahan lang tayo, that's how we designed those materials. So kung kayo medyo hindi kayo if you're not yet confident with your grasp of the research process, so meron tayong mga materials available. So we highly encourage you to follow us on Facebook kasi doon kami nagre-release ng materials almost daily. And then yung YouTube channel naman namin, doon yung makikita yung mga lectures na dinedeliver namin on a weekly basis. So we tackle fundamentals, meron kaming tutorials, but then we're also quite proud to say that over the past year and a half, we have been coming up with special topics in STEM. So kaparehas itong biology lectures natin. So what we hope that you take from this is not only yung learnings dun sa topics, but the sense of inspiration. So Professor Len, I actually took note of a couple of things that you emphasized repeatedly earlier, So I like the question that you asked the students. So ano ba yung mas mahalaga, traditional or modern techniques, And I think coming from practice rin po, I'm a chemistry major. It is always, so students, it's always very important for you to learn the fundamentals, yung traditional. Kasi yun yung basis ng lahat ng modern techniques. And Yes, and the most important thing is we have to make sure that yung continuity ng expertise natin dapat natatransfer yan from generation to generation. Kaya din namin ginagawa to. So, Filipino Science Hub is composed of Filipino scientists and educators from different parts of the world and trained in different areas. Bakit namin ginagawa to? Kapag kasi nangyaring puro influencer na at puro nagtitiktak na, mahihirapan tayo na i-sustain yung development ng Philippines. And, would you believe that the Philippines is lacking scientists right now? In 2018, ideally, dapat meron tayong 1,000, dapat meron tayong about 800 scientists per million Filipinos. We only had barely half of that. And para umunlad tayo, kailangan natin ng mga scientists. So hopefully, na-encourage kayo ng talk ni Professor Len to consider, biology and microbiology, as a career. Ayan. So, meron bang may questions? Parang nahihiya yata yung students natin.
Huwag kayo mahiya.
Oo, tayo-tayo lang to. Tayo-tayo lang to.
Tanong kayo. Kasi alam niyo, sinasabi ko lagi sa estudyante ko, yung mga matatalino, nagtatanong.
Opo.
Meron tayong tanong.
Ayan.
Natin. Pero bago po ang lahat, banggitin ko muna, according to Sir Jay, na teacher ng ating mga student audience today, from Bataan, sobrang thankful daw si Sir kay Prof. Len at sa Phil-Sci Hub, and this is a very memorable experience daw para sa mga students niya. Salamat daw po, ma'am. And hopefully, sir, makaka-attend ulit kayo ng students mo sa mga susunod na sessions natin ngayong Bio Month.
Yes, please, stick around.
So, acknowledge din natin yung
mga... sa Bataan.
Yeah. Acknowledge din natin yung mga students na nagpa-participate sa mga pa-quiz ni Prof. Len kanina habang nagdi-discuss. Ayan, very good kayo dyan. So, yung question, it's from Iggy Martin kanina. Lahat po ba ng bacteria na nakukuha natin is hazardous po ba? Kasi po nabanggit niyo po kanina na some bacteria are beneficial. Thank you po. He's from Bataan National High School.
Ulit yung first, ang narinig ko lang is some bacteria are beneficial. Sorry.
Oh, sorry. Mahina yata ang audio ko. So, lahat po ba ng bacteria na nakukuha natin is hazardous po ba? Kasi po, nabanggit niyo po kanina na some bacteria are beneficial din po.
Oo. Thank you, Jan, for the question. Thank you for your question. Kasi alam nyo, yun lagi yung naririnig ko, no? Okay, let's talk about microbes. Or let's talk about bacteria. Anong first impression nyo? Ang lagi nilang sinasabi, kakatakot. Kaya, magkakasakit kami. No? Laging ganun, ano? But actually, there's quite a lot of long list ng mga beneficial na microorganisms na nakakatulong. No, I think isang magandang topic pa yan, no? Na pwedeng webinar, ano? Who are the good bacteria? No? Meron nakakatulong sa atin para mas i-purify, linisin yung mga polluted water, polluted soil. No? Those are good bacteria. May mga mikrobyo tayo, hindi lang bacteria, no, na nakakatulong sa atin to produce metabolites para labanan yung mga disease-causing mikrobyo. Kaya siguro parang maghapon na huntahan tayo niyan pag ilista natin lahat ng beneficial. So isipin natin ngayon, pagkatapos ng Zoom natin, no, na so hindi pala lahat ng bacteria ay bad. So,
yes. Meron nga na kapag yung gut bacteria natin ay mawala siya, yung kahit magaling yung immune system mo, hindi siya gagana kung wala kang gut bacteria.
Tama yan. Narinig niyo na ba yun na we are more bacteria than humans?
Yes. Kasi yung immune system natin, nagre-receive lang siya ng signal from our gut. So kapag wala yung good bacteria natin, walang purpose ang ating immunity. Hindi siya gagana ng maayos. So ganun ka-importante ang field nito ni ma'am.
Okay, so kung dun sa webinar ni Dr. Connie Gibasa, Know Your Fungi, dito naman Know Your Bacteria.
Yes. Gusto ko din yung nagsasuggest si ma'am. Along the way, nagsasuggest siya ng mga topic.
Yeah.
May tanong ako.
Yes.
Yung
yes
good bacteria ba naging bad?
magiging like
Kailan siya naging
kaya
bad?
magiging bad siya.
Oo.
Alam niyo, meron tayong tinatawag na horizontal gene transfer, no? Minsan mabait naman talaga siya. Wala siyang kamuang-muang, lalangoy-langoy lang siya sa ocean, lalangoy-langoy lang siya sa body of water. But there are some microorganisms na nanggaling sa sewer system, no? Or kunwari nanggaling siya sa ating mga kanal, no? Tapos hindi tinreat, tinapon natin sa body of water, meron sila mga genes, no, na that makes them bad, no? So ang nangyayari, yung mga natatahimik na naglalangoy-langoy diyan sa dagat, no? Pwede siyang or capable siya na tumanggap nitong mga bad genes na to. So originally, kabait-bait niyang nilalang, pwede siyang maging resistant. At kapag na-expose yung mga fish o kaya humans sa kanya, pwede magkasakit. Kaya pwede talaga maging bad. No? Kaya iiwasan natin yun. Paano? Kailangan i-treat natin yung water bago siya itapon sa ating mga surface waters or bodies of water.
Para po pala yung siguro yung layman's term, para sa akin na hindi biology expert, para yung chismis, po. Meron mga nananahimik dyan, tapos pag nasamahan ka ng medyo machismis, nai-infect ka. So, ayan. So, ganoon din pala. So, so that's a very important insight po, kasi yung wrong notion about bacteria. So I think I myself grew up na 'di ba po parang magugas ka ng kamay kasi may mikrobyo. So 'di ba? Pero yung 'di ba Yakult for example, 'di ba po? Yan.
So pwedeng opportunistic.
Yeah.
Yeah, opportunistic. Meron din tayong opportunistic talaga.
Okay. Kasi meron kayong true
ng
pathway.
tamang panahon. Yan. Parang sa fungi, di ba, Mama Cons? No? There are opportunistic fungi. Nananahimik lang sila, pero kapag ikaw ay bumaba ang resistensya, boom, meron kang aspergillosis, meron kang candidiasis, and so forth. Yun din yung sa bacteria. Nananahimik sila dyan. Tapos biglang boom.
Yung mga tinatawag naming HIV person, sila yung parang human petri dish. Lahat tumutubo.
Meron pa tayong isang question, ma'am. From Carl Raymond Consuegra. Good day po. I'm curious to know what are the limitations po of using omics technologies like metagenomics, and how do they affect or influence studies related to microbial communities?
Siyempre, ang unang limitation dyan ay may pera ka ba, te? Yan talaga. Oo. Alam niyo.
Mahal nga. Kita ko kanina yung mga genomics eh.
Oo, kasi siyempre, galing ako sa Korea, no? So, ang bit-bit yung technology pang mayaman. Ano?
Meron na ba tayo dyan, ma'am, sa atin?
Right. So, pagdating ko dito sa Pilipinas, sabi ko, kaya ba namin yan? But you know, it's really, we are really very thankful how the Philippine Genome Center extends help, no, to scientists like us na, you know, mababang uri, mahirap lang. So they really extend help, kung paano namin magagawa. There was one time that they had this wonderful program that assisted scientists into whole genome analysis. Ang mahal kaya noon, no? Yeah. Pero they assisted, no, na pwede kang magkaroon ng whole genome analysis for free, tapos may training ka pa, bingo yan sa loto, no? And then right now, they're assisting small projects like us, under the PCHRD to have really yung rates na kayang-kaya ng iyong budget, ano? So halimbawa, ma'am, wala akong PCHRD project. Paano po yan? So meron din namang mga suppliers that connects us with sequencing facilities that gives us reasonable rates, no? So yun ang unang-unang limitation. Pangalawa, kunyari mayaman ka naman, ang dami-dami mong pera, no? Ano yung second limitation? The internet connection. Yan. So kailangan mong supercomputer to analyze such huge data. Yung mga estudyante ko dito, kaway-kaway ulit kayo na naghihintay ng 2 to 3 days bago ma-download ang aming data. Download pa lang po yun. Wala pang analysis, ano? But then again, sabi nga nila, yung mga anghel, lumilipad lang yan sa paligid mo. Kailangan mo lang silang ma-recognize. Meron po tayong DOST COAR o yung COARE. Sila po yung nagbibigay sa atin ng access para sa supercomputers nila. San ka pa? No? So ito, nagbibigay sila ng mga access. Yung mga estudyante ko can use that even at the comforts of their home using their own PCs, and we can analyze those particular huge data, no? So sa tingin ko yung mga taxes na binibigay nyo na napupunta sa DOST ay talagang gamit na gamit sa para sa aming mga scientists para ma-mentor namin yung aming mga estudyante with metagenomics. So wala kang pera, wala kang internet, nabigay na po natin. Tapos yung third talaga na is interest, no?
Yes.
Minsan, hindi sila interesado sa dry lab. Ma'am, gusto ko pa rin yung nag-autoclave ako. Gusto ko pa rin yung nandun ako nakalab ka, no? Na nandun ka sa lab, and so forth and so on. But right now, no, students are trying to appreciate now na meron pala, laboratory din pala yun, no? Gawa din pala yun ng scientists. Kasi because of the pandemic, most of the scientists became dry lab enthusiasts. Kasi wala tayong magagawa eh. We only have data in the internet. So kapag meron kanya ng 3 na yan, pwede na tayong mag-roll. And we have a community, a huge community of metagenomic analysts that can help you, at least assist you start. You know? So marami kaming natulungan na magsimula, na ngayon ay baby steps. Kaya natutuwa po kami na ang Pilipinas ngayon ay marunong ng mag-metagenomic analysis.
Yan. Gusto ko lang din i-point out yung kanina pa nababanggit ni Ma'am. Sa mga, lalo ngayon, I think right time para mabanggit sa mga kabataan kasi mga senior high school na sila. I think importante yung mga nababanggit ni Ma'am na field ng biology ngayon since na-open na rin niya yung genomics, bioinformatics, NGS, and next generation sequencing, and all other types of sequencing. Marami pang iba, even sa medical field, ginagamit siya like single cell RNA sequencing. Ito kasi ginagamit siya sa pag-identify ng mga bagong bacteria. Kunwari, ito yung mga ginagamit ni Ma'am kanina. Napakadaming opportunities for both sa studies abroad and jobs sa bioinformatics and programming, tsaka management ng big data. Kasi ito yung mga single cell, itong mga RNA sequencing natin, kunwari, or DNA sequencing, napakalaking data niya, as in, hindi mo mai-imagine kung gaano kalaking data yung na-generate niya. At useless siya kung walang skilled na person na mag-process ng data. Kasi, kunyari, as a scientist ako, hindi ako maalam kung paano yung programming, hindi ako marunong kung paano gumawa ng, paano mo i-convert yung data, yung numbers na na-generate mo from your lab, from sequencing, into an interpretable data para sa maging graph siya or para maging plot siya or something, graph. So hindi ko siya maintindihan unless gagawin siya ng bioinformatician, naka-collaborate namin. Ganun siya ka-importante.
And I would like to raise the...
Comment lang ako kay Janice. The way to go really is collaborate. No?
Yeah.
Now, the team is now a multidiscipline team.
Yes.
Hindi kaya na microbiologist lahat yan. Yeah. So we really need to accept the fact that we can only do so much. Kaya sa team namin, meron kaming taga-comsci, meron kaming chemist, meron kaming civil engineer, meron kaming chemical engineer, meron kaming geneticist, bio. Hindi mo kailangan aralin lahat.
Yeah. Meron siyang kakayanin mo ding aralin. Kasi ako, chemist ako by profession. Tapos napunta ako sa nagsi-single cell RNA sequencing. So, hindi ako maalam sa mga ganon. So, kailangan mo ng person na bioinformatician na trained talaga siya na mag-process ng mga ganon kalaking data.
Yep.
Pero it doesn't hurt kung may chance kayo mga estudyante mag-aral ng C++ o di kaya Python, yung basic.
Yung mga ganon, Python, mga R.
Yung mga available sa PC na kailangan hindi yung katulad sa Mac na pindot-pindot lang. Pero kung sa PC ka, kailangan mong magganyan. Di ba, Doc Jeff?
Yep. Gusto ko lang din pong erase yung point. Kasi ito, marami po kasi tayong high school students in the audience. Yung notion sa high school na biology is mostly memorization. So clearly, napakita ni Professor Len that it's not all about memorization. There's a lot of cool things that you can do. And one very important thing that you guys have to know by now is that this century and the upcoming century is a century for biology. Most of the transformational technologies happening, yung innovation and yung magiging future natin will be driven by developments in the area of biology. So you have to take note of that. So in terms of in terms of the skills needed, not only in the Philippines but globally, it's biology.
Yes,
So,
but
yeah.
As a microbiologist myself, a mycologist, I would like to emphasize na kailangan aralin nyo ang chemistry.
Yeah.
Everything is chemistry in microbiology.
Yep. So that's a form of So alam nyo, ang ganda po nito. I'm really enjoying this discussion because Prof. Len, thank you so much for a fairly solid and democratized content po. And then this essentially is a very good platform for us to have this discussion kasi baka kasi yung mga bata you're trivializing biology. No, it's one of the coolest things out there right now. Yan. All right. Questions pa?
Meron pa dito.
Yep.
From Lindsay Ann. Since nabanggit po na may good bacteria that turns bad, does bad bacteria turn good? Akala ko parang tongue twister.
Kailangan basbasan siya ng holy water.
Ang gala ng paa.
There are some bad bacteria, kaya sila bad because they really need a host to survive. And without those particular hosts, nandiyan lang sila sa palipaligid. In that state, mabait sila. Pero pag na-meet na nila yung host, hindi na. So, kumbaga, yun talaga kasi yung nature nila. And without the host, they may not even really survive. Parang intriguing yun na. Mabasa nga yan. Hanapin ko yan kung paano maging mabait ang isa.
Meron ba kasi, hindi ba parang meron lang na balance ng good at bad? Importante yun.
Kailangan talagang balance. Kailangan
bakit nandun yung bad?
Yeah. Sa fungi, kahit very good yan, kahit yung mushrooms, kung dadapo yan sa mga immunocompromised, magiging bad siya eh.
Oo. So there are some states talaga na convert talaga sila into something bad. If kailangan nila, kakain yan eh. I need to survive. So anong pwede kong kainin dito?
Ayan.
Pero tayo rin, di ba? We adapt. So it's up to you whether you turn good or not.
Ay, ito, may
maganda itong question na ito, Jack.
Question.
Oo. May I read it? So sabi niya, ito po, this is from John Vincent. Ay, natitrigger na yung students natin. Ang dami nila.
Oo.
So John Vincent, diba? Ganun naman kasi talaga yun. Ayaw na lang sila yung mauuna, no? Pero pag nasasagot na yung mga tanong, yan, diba?
Oh my God, ang dami na pala.
There you go. Don't be scared to ask. Alam nyo,
3 hours tayo dito.
Ang lagi naming tips sa FSH Hub, whenever we engage with students, alam nyo, ang laki ng mapapala nyo kapag makapal ang mukha nyo. Fortune favors the brave and the prepared. Hindi lang matapang, no? Kailangan medyo handa ka naman. Anyways, moving on to the next question. So, John Vincent Revocal. So, ang tanong po niya, I think, pwede ko itong itanong kasi Mama Connie is here. I think she can also answer this. Ang tanong niya is, ask ko lang po if nakadiscover na po kayo ng new bacteria. So if opo, paano niyo po sinisigurado kung ang bacteria na na-discover niyo po ay hindi pa po nag-i-exist talaga in the entire hindi pa siguro po na-discover na siguro yung gusto nyo itanong. Kasi po marami pong lab din, paano nyo po malalaman na unique po talaga ang bacteria na discover nyo. Siguro ito, lagyan natin to ng caveat, no? Kasi we have an expert sa bacteria, which is Professor Len, and then we have Mama Connie, who is also, who is an expert in mycology. So paano po ba? Sa experience nyo po, let's start with Professor Len.
I love that question, although hindi ako authority kasi hindi ako taxonomist. Sa bacteria, usually malalaman mo naman kung ano, kaya pinaliwanag ko sa inyo yung yung from the colony to microscope to physiology, hanggang sa ma-identify nyo yan. Kasi meron yan checklist para malaman mo kung bago or hindi. And you compare that with the database ng taxonomy. Parallel yan with the fungi. So I think mas masasagot ni Mama Connie yun kasi mas expert siya sa taxonomy than me.
And teka, before sumagot si Mama Connie, just to give you guys trivia, Mama Connie, ilang species na ng fungi ang na-discover mo?
Yung mga 20?
20. Take note, Filipino scientists discovered 20 fungal species. And then ano nga po yung next domain ba yun, Mama?
Order at saka
order. Or diba, hindi talaga ako biologist. I'm so sorry. Ilang orders po ng fungi ang, para order lang ng pagkain, ilang orders po ng fungi ang na-discover nyo?
Isang family, isang order, 4 na genus.
Yan. So, take note, everyone, so to all Filipino students out there, you can actually be like our panel of biologists. So makaka-discover din kayo ng species on your own. Ayan, Mama Connie, please answer.
Okay, so like bacteria, so sa bacteria meron silang gram stain. So you will start with positive and negative. Sa amin naman, kasi ako clinical mycologist. So ang major na focus ko ay yung nagko-cause ng disease. So it's in the Ascomycota, which is in the subkingdom Dicaria. So i-look up nyo to para kasi wala tayong time para mag-discuss. Maybe in a later seminar. Anyway, under the subkingdom Dicaria, merong Ascomycota at saka Basidiomycota. So yung Basidiomycota, yun yung mga mushrooms. Yung Ascomycota, yun yung mga hindi mushrooms. Okay? So, meron akong test dyan. So, before everything else, so ang fungi, meron siyang structures na kailangan namin makita, like yung spore or conidia ang tawag sa fungi. Kung wala kaming makikitang ganon, sterile siya, wala kaming basis para i-identify siya. So ito yung traditional hindi yung advanced. So ang gagawin namin, ipapagrow namin siya sa isang medium na magdi-differentiate between basidiomycetes, yung mushrooms, at saka yung hindi mushrooms. So kung hindi siya mushroom, then we will go, for example, yung gram-negative, meron ka ng schema doon, meron ka ng flow, kung anong susunod mong gagawin. So sa amin naman, papatubuin namin siya, bibigyan namin ng maraming pagkain, o di kaya i-stress namin sila para kailangan nilang mag-survive para mag-produce sila ng structure. So kung makakita na kami ng structure, doon na kami magsimula, okay, meron siyang ganito. Ika-classify na namin sila by family, kung kaya ng genus. So, from there, sa genus, kung kaya mong i-differentiate by morphology, pwede. Pero ngayon kasi, halos pare-pareho ang itsura in the same genus. Tapos, kasi kami sa clinical, wala kaming time maghintay ng 2-3 buwan para mag-produce sila. So ang ginagawa namin yung advanced na technique, which is DNA sequencing, proteomics like MALDI-TOF. So i-look up nyo din yan para malaman nyo kung ano yon. Or yung mga ibang tests. So kung makita na namin sa DNA sequencing, okay, nakita na namin, ito yung species niya. Babalikan namin ngayon yon. Papatubuin namin kung para doon sa genus na yon, yung grupo na yon, kung anong kailangan niyang medium or yung culture medium para tumubo siya, para mag-produce siya ng structures. Tapos pag nagawa na siya ng structures, i-compare namin sa ibang species under that genus. Kung kakaiba siya, ibig sabihin, wala siyang kapareho doon sa genus na yon. So ngayon, sasabihin na namin, okay, kakaiba to. This is distinct from all the other species. Mag-sequence pa uli kami ng marami pang genes para mas marami, para mas maganda yung data namin. Para masabi talaga namin na this is unique. So doon kami pwedeng magsabi na this is a new species. Tapos ipapublish mo bago ma-accept na may bago kang species. Kung yung mga experts na magre-review ng paper mo, sasabihin nila, okay, you're good. Kaya mapapublish na yung bago mong species.
So it's a combination still po of traditional methods and modern methods. Ano po?
You have to know your fungi.
Ayan. So, dalawa na yung tatandaan. You have to know your bacteria and you have to know your fungi. And by the way, merong webinar si Professor, si Dr. Gibas on fungal mycology. And she talked about discovery then to a certain extent na po. Yan. So check it out dun po sa Phil Science Hub YouTube channel kasi nandun yun. All right. So perhaps we can entertain a couple more questions para naman we don't overlap with their tanghalian. Ayan.
Chikahan to,
Kapit. Ito pa ba?
Apo. Ayan. So which ones would be good?
Meron pa from Justin Punzala. Ito kasi isa sa mga advocacies ko din. Yep. Hello po, regarding po sa resistance of other bacteria to antibiotics, would there be a way to lessen their resistance to it?
Yes.
Interested siya sa antibiotics.
Resistance to antimicrobials actually resulted from yung hindi natin tinatapos, yung ating mga antibiotics prescriptions, Pero 7 days yung pag-reseta sa'yo, 3 days pa lang, medyo gumagalaw ka na, ayaw ka nang tapusin. Yan. O kaya, there are some issues of these antimicrobials present in nature, like in soil, water, etc., that seems like the pressure, the microorganisms there to adapt to it, so nagiging resistant sila, and so forth and so on. So, paano natin maiiwasan itong mga ito? So, kung ikaw ay naresetahan, yung doktor, complete the course. Complete the whole course of the prescription. In that way, alam mo na makukumpleto yung pagpatay mo dun sa target community population ng pathogen mo. Tapos yun na, yung pangalawa, we really need to check out the wastewater right now. Kasi marami na tayong problems. Sabi namin ng new generation of pollutants. Ito na yung mga antimicrobials and other molecules which pressures microorganisms to mutate. And then, because of those mutations, pwede siya magkaroon ng new characteristics that we cannot imagine right now. Anong effect sa atin. So yun nga, maiiwasan naman ang kumbaga, merong mga papers na nagsasabi, inherently, nandyan lang yung resistance gene sa kanyang DNA. But it would really take the environment to turn it on.
Yes.
So kung hindi naman nape-pressure, ang mga mikrobyo na to because of those pressures na antimicrobials, mga heavy metals, at kung ano-ano mga chemicals, it won't turn on the genes. Pero kung nandyan yan, because of pollutants, because of yung kagagawa natin na hindi pagtapos sa antibiotics, mapapressure sila mag-attack. And that give rises to the resistant microbes.
Siguro po, next question, at I think this is something na magandang ma-address. So this is from Junel Maureen Ignacio. So her question says, hello po, sa tingin ko, matagal na po itong itinatanong ng lahat. Why do antibiotics only treat bacteria and not viruses? Thank you po.
I don't know why antibiotics
only treat bacteria and not viruses.
Bakit ba? Bakit? Oo nga, kasi di ba galit na galit tayo. Meron ka trangkaso, ba't ka na antibiotic? Kasi it's mainly because of the principles of antimicrobials. Antibiotic, antibio, antibuhay. Wala naman po kasing buhay yung viruses. Kaya hindi talaga sila tatamaan ng antibiotics. Ang tinatarget nila ay cell wall. Wala pong cell wall ang viruses. Or minsan may mga target silang mga organelles. Wala pong organelles ang ating mga viruses. So that's basically the reason why antibiotics won't work for viruses. Alam niyo yung mga viruses nga, pahinga lang yan Pahinga lang. So, at the course of your bed rest, hydration, tamang nutrition, gagaling ka kasi iintay nyo lang kung matapos yung kanyang life cycle, 7 to 14 days, or sa ibang, of course, sa ibang cases mahaba, no? Ang nangyayari is because napakababa ng resistensya nyo during the time na dumating yung virus, ano, natamaan ka talaga. At kung mababa pa yung resistensya mo, meron ka tinatawag na comorbidity. Ito yung mga echoserang bacteria na, uy, mahina na siya because of the virus, ano. At yun, papasok, they will come in. That's why, because of that situation, binibigyan ka ng antibiotic. Not directed to the virus, but the comorbidity. Example, nung nagkaroon ng COVID, nagkaroon tayo ng mga swine, ng mga influenza outbreaks noon, ano, after nun, sumunod yung pulmonya, no? So sabi namin, bakit nagkakaroon ng pulmonya? So parang ganito po yung analogy, no? Yung virus, pumasok siya dun sa red carpet, no? Tinabas niya lahat ng mga nakakasukal ng daan. Aba, pagdating ng mga bacteria, VIP na sila, latag na yung carpet. It's very easy, no, for them to make you sick or more sick, mas malala, ano. That's why antimicrobials yung tinitreat. Pero yung idea ng iba na mag-antibiotic ako kasi meron akong sipon, which is a viral disease, or trangkaso, ay hindi po kayo gagaling. Mas lalo yung pinapahirapan yung katawan nyo because yung mga ibang bacteria na tumutulong sana sa inyo para labanan yung inyong situation, your good bacteria inside your body, ay pinapahina nyo din kasi sila yung nata-target nung ininom yung antibiotics.
Siguro po, ako, I would take this opportunity to ask a couple of questions because we're about to wrap up na, no? So, siguro po, Prof. Len, could you make a comment po siguro, ano po yung situation ngayon when it comes to yung bilang ng microbiologists in the country and yung gaps sa expertise and sa skills na kailangan natin for us to be able to keep up with our neighboring countries kasi ang bilis-bilis ng development, no? So yun po, could you make a comment po?
Oo. Buti na tanong mo yan. Gusto ko talaga i-shameless plug yan. Ano?
Opo. And that's part of
during the
this.
pandemic,
Yeah.
halata nila na kulang.
Yeah.
Kulang ang microbiologists, ano? Microbiologists helps the doctors, help the other people na in the health sectors to know and identify this virus. Paano sila aralin? Paano magmalaw yung virus na yan? Ano bang gagawin nating bakuna dyan? Anong gagawin gamot dyan? You have to know the target. Kailangan mong kilalanin ng kalaban mo para makadevelop ka ng cure, makadevelop ka ng iyong panlaban, no? And because of that, biglang nagulantang, no, ang society na wala tayo masyado microbiologists dito, ano. And we are actually awaiting, no, in the Philippines to professionalize, no, the microbiology as a profession dito sa Pilipinas. Ito ay isang bill na inintay na lang ng pirmahan nito, no, sa joint congress, no. And after that, magkakaroon na rin tayo ng PRC exam.
Ay, grabe.
Wow.
Magkaganon po ba sa...
Sabi nga na, para saan yan? Ngayon, meron na tayong VIP o yung Vaccine Virology Institute of the Philippines. Kailangan ng microbiologist dyan. Meron tayong food industry. Kailangan ng microbiologist dyan. Pharma, kailangan ng microbiologist dyan. Teachers, kailangan namin ng microbiology sa institutions like your higher education institutions. At marami pang iba. Ang dami nating diagnostic labs na hindi micro ang gumagawa. Nakakaloka, di ba? That is why we at the Philippine Academy for Microbiology is really working hard, no? At ang una namin ginawa is to harmonize, no, the syllabi of the microbiology courses sa lahat ng HEI sa Pilipinas para pare-pareho tayo natututunan. Pagdating natin sa PRC, pare-pareho tayo ng sagot. No? So, kailangan natin ng marami. Yes, Jen.
And hopefully po dumating yung panahon, ma'am, that like where we are producing microbiologists not only to cater to yung existing industries, ano po, yung incumbent needs, but then hopefully instead of hopefully yung microbiological society natin ay we can work at the offensive na we are developing technologies driven by microbiology, because just to give you an idea po, in the US, microbiology, yung microbes can actually carry out processes and produce materials na mahirap gawin outside of a living cell. So ganun na siya. So natatransform na rin yung ibang industries by microbiology. So I'm hoping na sa mga bata nakikita niyo yon. Forward thinking, we're not only gonna cater to the existing need. Kailangan meron tayong enough manpower para ma-drive talaga yung technology development. So sana po Professor Len, we hope that siguro a good percentage of the students participating in this Zoom session would pursue microbiology as a career, kasi that's the future. So I am telling you guys. Yan.
Ang dami-dami pong gagawin. Ang
Yeah.
onti-onti po namin. No? Kaya I'm
Yeah.
really very thankful sa UP Manila for opening the microbiome track for the BS Bio program. Maraming salamat po. Yung iba dito, nandito yung mga estudyante namin sa micro track. For the other institutions, especially now yung mga students natin ngayon na nag-iisip, ano kayang kunin kong course?
Apo.
Microbio po tayo. Pwede niyong kunin yon through the BS Biology programs of various universities. Siya po ay micro major. No? Pwede rin kayong kumuha ng BS Micro. Ang UST po ay merong microbio na program. Kung kayo naman ay nakagraduate na ho ako ng BSN, paano yon? May MS Micro po tayo. Pwede kayo magpatuloy, di ba? Or naka-MS ka na, may PhD Micro pa po tayo and other courses. So it's really a very inviting year for us. Medyo napapansin na po tayo because of COVID. Para nagpasalamat kay COVID kahit na ang sama-sama niya. Pero because of that problem, we have identified that we really need more microbiologists in the country right now.
Okay. Sabi nga ni Prof. Len, be one of us. Yan. Yes, Mama Connie.
Pagma-micro,
be one of us.
bahagian nyo naman ang mycology.
Yan.
Kasi, ang mga mycologists, nagiging dinosaur na, namamatay na kami. Wala na, nobody's
mycology.
Yeah. And
microorganisms. Ayan. By the way, ipa-plug ko din, when our lab reopens, kasi nag-reopen pa lang ang mga laboratory ngayon because of the pandemic, we're very happy to open it up for junior, senior high school, for your internship, or yung
ayan.
po kami. You can just send us an email. I think I gave my email. And I can also put it here in the chat box. Sabihin nyo lang po, kasi we're very happy to let you in our lab and train about microbiology for health and environment.
Ayan. So, grabe, that's very exciting. So, siguro po, yan, ito po, meron ako nakitang question po earlier, and we can actually make this the last question. Yung question po kasi kanina was, as a biologist or as a successful microbiologist, ano daw po yung challenges that you experienced, and then how did you siguro po surmount those challenges?
Ito ba yung as educator during the pandemic? Yun ba yun?
Hindi po. As a biologist po yung tanong.
Challenges.
Apo. Siguro not as an educator, but as a researcher siguro. Baka po ganun yung tanong.
So, syempre, iba-iba kasi yung challenge kung nasaan ang locations. Iba-iba rin yung challenge kung ano yung gusto mong specialization na nagagawin. So I can only answer for myself. I'm sure the other microbiologists would have a different story. So ang challenge sa akin nung nag-uumpisa pa talaga ako, ang hirap magpapansin sa funding, walang laboratory, walang space, walang gamit, walang ganon. Pero do not lose hope. You always welcome networking. You always welcome linkage. Huwag kayo mahihiya na ito lang po yung meron ako ngayon. Pwede bang makitrain muna dyan? Because of that training exposure, malalaman mo na kung paano mo uumpisahan yung sayo. no? Kaya that's why I'm really very excited na makakapag-train kami ng mga tao kasi nagsimula kami sa nothing sa UP Manila, no? Na nakikilab kami sa UPLB o nakikilab kami sa kung saan-saan man. But right now we're opening our doors, really excited. The main driver na lang sa inyo talaga is your interest, no? We really would like to see, and if there's this tiny spark of interest in you, go at sundin nyo yon, sabihan nyo lang kami, no?
Yeah, sabi nga ni Prof. Len, it's yours for the taking, no? So, it's good to know po that yung landscape sa atin, yung infrastructure, it's up and coming, you know? So, that's very encouraging. Grabe, sobrang iba na po kumpara nung nasa Pilipinas pa po siguro kami.
Ang layo na lang nararating.
Yan. And so, ito po, siguro I would share this screen kasi
last line, no? Wag niyo isipin na dahil nasa Pilipinas kayo ay wala na tayong magagawa. Meron yan. No? Laging meron. Isipin niyo na lang palagi is, sino ang tatakbuhan ko muna? Sino ang pwede mag-mentor sa akin? Saan ako pupunta? And then, pagkatapos kong matrain, balik kayo dito. No?
Yep.
Yung opportunity at oras para kayo naman yung magbibigay ng opportunity para sa inyo.
All right. So if you guys can see my screen, ito yung statistic na gusto kong i-share sa inyo. So in 2018, UNESCO did a study, and their recommendation was for the Philippines to be able to keep up with its neighboring Southeast Asian countries, no? Hindi pa ito yung mga first world countries sa buong mundo. We need 380 scientists per million Filipinos. That year, disappointingly, we had 189. Hindi pa tayo nangalahati. So we have to work on this. And in that year, if you would compare Singapore, with its very small population, it had 6,900 per million Singaporeans. Malaysia had 2,100 scientists per million Malaysians. Thailand had 975 per million Thai. So sa mga high school students natin, kailangan natin ng napakaraming scientists kasi kailangan yan para sa sustainable development. Ayan. And then, lastly, before we end the session, I would just like to share with you, ito yung sinasabi kong portfolio. So, it's already out there. These are Tagalog English training courses on research fundamentals. So we have covered all of the steps of the research process. And then, etong talk ni Professor Len will be part of, our special topics portfolio. So, yan, ang dami nating areas na fini-feature dito. So, all foundation science courses, so physics, chemistry, biology, data science now. We also have featured scientists and experts from allied STEM area. So, marami kayong mapupulot dyan. So, and the reason why we're doing all this is to essentially spark that sense of inspiration and interest sa mga high school students kasi kailangan ng Pilipinas more than ever ng maraming STEM professionals. Ayan. So, do we have
Kuya Jeff, before tayo mag-end po pala, may mga nagtatanong about dun sa virtual lab po na ginamit ni Prof. Len kanina. Ano daw po ba yung virtual lab kanina? Yung may...
Yes, I just have to check it out sa kabilang computer na. But probably I can share sa Philsci Hub and then you can,
Yeah. So, punta po kayo din sa Facebook page. We will post the link. Ayan. By the way,
Jeff, the link is there where they can go.
So we'll extract that from the slide. So by the way, we would like to welcome JP Onya. So JP Onya, by the way, is the Vice President of Filipino Science Hub, and he is the head of our most successful portfolio so far, which is Research University. Kaya meron kayong special topic because of Research University. And he is dialing all the way from Finland. So JP, good morning.
Hello, good morning po. Mag-hi lang ako sa mga estudyante natin. I think marami yung mga taga-Bataan. I worked in Bataan for 7 years. So alam ko yung yung province. So ayun po, nabanggit ni Kuya Jeff yung portfolio of topics sa Research University. You can just go ahead and go to www.philscihub.com to check out all of our courses, and we are issuing certificates of participation kung na-take niyo yung mga courses na yun. And speaking of certificates, I would like to present a certificate of appreciation to Professor Marilen Balolong of the Department of Biology, College of Arts and Sciences, UP Manila, for delivering the webinar Evolving Strategies to Identify Functionally Profile Bacteria from Communities, organized by the Filipino Science Hub and held by Zoom and YouTube Live on the 2nd day of April. Signed yours truly and our CEO and founder, Dr. Jeffrey Bumkin. Yep. And also, speaking of certificates, we already posted the Google form for the evaluation at yun na rin po yung mag-i-issue, magsisilbi po na platform para ma-issue yung mga certificates nyo. So make sure that your email addresses are correct and accurate dun sa Google form kasi hindi namin masasend yung certificates kung mali po.
Yep.
Ayun, and all of your personal details.
Yep, and
so there.
Please be patient po kasi we generate certificates and batches po, no? So it might take a week or two for you to get your certificate. So medyo kami po kasi ay team ng volunteer. So hindi po namin ito full-time job. Kami po ay meron po kami full-time job on top of this. So anyways, so Professor Len, siguro po final message para po sa students and educators natin who are in the audience in this session.
Jeff, kinikilig na nga ako sa mga message nila dito sa ating chat box,
Opo.
For the students who are here right now, alam ko malayo pa yung tatahakin nyo, Kagaya nyo noon, hindi ko pa rin alam yung gagawin ko. Kagaya nyo noon, gulong-gulo pa rin ako at parang ang hirap-hirap ng mga bagay-bagay. It's really very challenging. Kagaya nyo noon, siguro parang sinasabi ko sa sarili ko na mararating ko kaya yung mga gusto ko, Continue to ask questions because of that questions, yun yung magdadala sa inyo to have the answers to those questions, Continue to be curious. Yan. Sabi ko rin sa mga estudyante ko palagi, keep the stupid ideas because sometimes those stupid ideas ay hindi na stupid in the next 5 years because of your probing. Kasi sa daming gusto mong sagutin yung tanong, So wag kayong tumigil magtanong. Wag kayong hihinto magtanong para maging aktibo yung brain niyo, And of course, always take partnership, collaboration, very very important. Tanggapin niyo sa sarili niyo na hindi niyo kaya mag-isa. It's not all about me to answer the big problems of this country or the world, but it's going to be together that we can serve the world. So through our science, through your skills, through your talents, kung ano man yan, ilagay nyo lang yan sa pool and then we will be using all of them to serve the nation. Thank you very much.
Yeah, ayan, very inspiring. So alam niyo po ako I'm hoping that the students are actually getting the message. So, you will succeed in life, but what would make your success very meaningful is if you share it, whatever it is, success or talent, higher meaning if you share it. And yung love for the country, sana hindi nawawala yun. Ayan. So, maraming salamat po, Professor Len. Everyone, this is just the first installment of 4. So meron pa tayong 3 pagsasamahan para sa Biology Month. And sabi nga po, so JP mentioned earlier that we shared the link to the Google form. So please let us know of your feedback. Tell us how we can improve. And then at the same time, may post test doon. So titingnan natin kung nakinig talaga yung mga students. Tapos susumbong ko kayo kay Sir Jay. Joke lang. Ayan. So, maraming salamat po sa pagsama sa amin. Yeah. And then next week uli. Yeah, we have another session. Ayan, maraming salamat po.
Kids, uli.
Opo, ayan. Siguro po, Professor Len, baka po pwede namin kayo ma-invite sa very quick debriefing after this. Separate session, we'll ask Ma'am Connie to share the Zoom link with you. But then, everyone, maraming salamat po. Kita-kits uli next week. Bye!
Bye-bye!